All right, here we go. Keith, you can go ahead. Yeah, if you're ready.
Thanks, Ryan. Thanks, Todd. Welcome, folks, to the Biogeochemistry Working Group session of the CESM Workshop. As you're probably seeing in the pop-ups, this meeting is being recorded, and that also includes the chat, so that questions in the chat will be able to refer back to them later after the meeting is over. We're going to, while I'm telling you some logistics here, I'm going to show another slide here about some of the principles that we try to adhere to, and we want all of our participants to follow regarding having a respectful dialogue amongst participants in our meeting. To I don't need to read this to you, you can read it yourself, but these are principles that everyone should be adhering to. After we have a handful of presentations covering a wide range of topics, we're going to have a discussion session where we're going to break out into Zoom meeting rooms, and that's going to be done randomly, and we're going to have some questions prompted to prompt discussion, and I'll get to those at the end of my presentation, which is going to come up in just a moment. Before we, that went a lot faster than I thought it would. I do want to introduce the other co-chairs of the working group. Gretchen Keppel-Alex from the University of Michigan and Abby Swan from the University of Washington. Thank you to them for your help with setting up this meeting. Are there any questions from the group before we proceed? And Todd, did I miss anything of logistics that I need to cover?
Sounds good, Keith.
Great, thank you. Okay. I'm going to go ahead and give an update on some activities of the working group. And I guess one thing that I did forget to mention is that we have 20-minute slots for all of the presentations, and we would like everyone to leave at least 3 minutes for questions at the end of their talk. If you're able to give more, then that would be great. And Gretchen is going to remind you as you approach those time limits.
Yeah, I'll give folks a reminder at 15 minutes. So you have a couple minutes to wrap up your talk, and then 17 minutes so we can reserve some time for Q&A.
Great, thank you, Gretchen. So I'm going to go into some updates. A lot of this material is repeat from what was presented on Monday. So apologies to those of you that if you get bored by it, but it's better to see the important stuff more than once, and there is some additional stuff in here as well. So one of the key activities of the Biogeochemistry Working Group is a coupled carbon cycle version of CESM, where atmospheric CO2 is being predicted by the model, and transient experiments are CO2 emissions based experiments. So CO2 fluxes are being prescribed from fossil fuel consumption and cement production. And then CO2 is transported by the atmosphere, and the surface components take up CO2 as air CO2 fluxes and respiration and photosynthesis and respiration in the land. And an output of that configuration of the model is having this prognostic atmospheric CO2. So these first plots that I'm showing are globally averaged annual mean surface CO2 in these ESM HIST experiments. And I'm showing the global average in observations as the black line. And this is the same curve that is used to force the model when we are not running with a coupled carbon cycle. The dashed line is showing this simulated atmospheric CO2 in CESM1 BGC, and you can see the growth of CO2 in CESM1 was too high growth compared to the observed growth rate in the atmosphere over the latter part of the 20th century. Particularly in the bottom panel where there's the model minus observations, you can see the slope of that dashed line is positive, indicating this bias in growth rate in the atmospheric CO2. And that continues on into the 21st century compared to the IAM models that are used to construct future scenarios. There are a couple of colored solid lines also in these plots, which are two ensemble members from CESM2 with this coupled carbon cycle model in this ESM HIST. When you look at the absolute values themselves, the curves are pretty much on top of themselves. It's hard to distinguish them. You do see some variation in the ensemble members when we look at the bias plot in the bottom panel. And there's a few things that come out of this bias plot that are worthy of note. One is that in the 19th century, around 1850, our coupled carbon cycle model produced atmospheric CO2 concentrations that were slightly elevated compared to observations. And this was a consequence of remaining drift after doing some spin up in the component models. The land and the ocean model both had drift of the same sign in CESM2, and they both were emitting carbon into the atmosphere. The drift was small, but they were of the same sign of releasing carbon into the atmosphere. And then that led to a slight increase in atmospheric CO2. And then when that CO2 became elevated, there was a negative feedback, which then changes the sign of that drift. And we end up getting a stable simulation with a slightly elevated atmospheric CO2 level. In CESM1, the drifts in the land and the ocean after doing spin-ups were of the opposite sign. And we ended up having a continual transfer of carbon from the land to the ocean while leaving the atmospheric CO2 relatively stable. So the fact that the atmospheric CO2 was slightly elevated in the 1850 is pretty much, or that it was stable in CESM1 and elevated in CESM2 is mostly just a byproduct of the remaining drift from the spin-ups. It's not something that we were able to, you would have easily been able to control from doing those spin-ups. The next feature that comes out in this bias plot is the slope in the mid-20th century in the bias, showing that the models are accumulating atmospheric CO2 compared to observations. This is pretty much unchanged from CESM1 to CESM2. And this is a feature that is present in many different CMIP class models. And it's an ongoing research to understand why most coupled carbon cycle models are not able to reproduce the atmospheric growth rate of CO2 in the mid-20th century. Now, in the latter part of the 20th century, the CESM2, we've built up a bias from having that bias in the pre-industrial value plus the extra accumulation over the mid-20th century. We're now about 15 ppm too high. But at that point, that bias becomes relatively flat. And one interpretation of that is that the growth rate of atmospheric CO2 in the model is matching the growth rate in observations. And this is a notable improvement from CESM1 to CESM2, where you can see that the growth rate in CESM1 was substantially higher than what was going on in observations. This improvement in the model is primarily due to improvements in CLM, from CLM4 to CLM5. Another diagnostic of the coupled carbon cycle is looking at the seasonal cycle of atmospheric CO2. And I'm going to show some figures of that from Barrow, Alaska, and Mauna Loa, Hawaii. The dashed line, just like the previous plot, the solid line is the observed seasonal cycle. And at Barrow, Alaska, there's a substantial around 15 ppm peak to trough difference in CO2 in the surface in observations. And in CESM1, we were that the amplitude of that seasonal cycle was low compared to the observed seasonal cycle. And that has improved considerably in CESM2. The simulation is not perfect. There's some phasing difference in the model compared to observations, but the improvement in the seasonal cycle amplitude is a notable improvement that we're happy about. And then the bottom plot is showing the amplitude of that seasonal cycle as a function of time, showing that if we just had looked at the early part of the 20th century, around 1920, CESM1 and CESM2 don't look that different. But as we progress through the 20th century, CESM2 grows considerably more than CESM1. In where we have the observational record at Barrow, CESM2 is doing a decent job of capturing the growth rate of this seasonal amplitude. So this is also a notable improvement compared to CESM1. But not everything about the model is perfect. If we look at Mauna Loa, Hawaii, and sample the model at the same altitude that the observation station is, which is up in the free troposphere, we do see that there is an improvement from CESM1 to CESM2 with the dashed line to the solid colored lines, but we're still weak compared to observations. And there's some ongoing research to understand if this is from surface fluxes or transport in the atmosphere to separate the bias into how much is due to those different aspects of it. So even though there are these improvements in the model, there still are aspects of the model that further improvement is possible. The next slide, I'm showing some atmospheric air-sea CO2 fluxes compared to a neural network filling a global map of air-sea CO2 flux, and the model does reasonably well compared to this observationally based product for the annual mean plot in the maps. And then the zonal mean with the seasonal cycle, the model is doing reasonably well. And the zonal average also looks pretty reasonable compared to this observationally based product. This is the product of Landshutzer. I realize I'm running a little short on time, so I'm going to pick up the pace here. One aspect of the ocean simulation that is a sore spot in the model is the deep ventilation, the ventilation of the deep Pacific. So if you look in this bottom row, this is radiocarbon simulated by the model. In the first column is delta 14C in the model, and we are actually off the color scale shown here. On the middle column is an observational product, and the right column is the bias. And the model is ventilation of the deep Pacific is too weak compared to observations considerably. And so this leads to excessive decay of radiocarbon, which indicates that this sluggish ventilation. And this has implications for global nutrient cycles because we tend to accumulate nutrients in the deep Pacific. And we had to put in some kludges to avoid oxygen from really going totally off the rails in our CMIP simulations. So this is a substantial deficiency in CESM2 compared to observations. Moving on to different flavors of activities of the biogeochemistry group is looking at analysis of simulations of the carbon cycle. And this is looking at simulations where we typically do in the climate model, a doubling of CO2 where the radiation sees this doubled CO2 and the surface components see this, the BGC surface components also see this increase in CO2. And there's some experiments here where just the surface components in BGC see this increased CO2. And it turns out that the terrestrial system seeing this additional CO2 leads to a warming that explains as a non-trivial fraction of the warming that you see in the full-blown doubled CO2 experiments. So what you see in this top panel is so this is the physiological contributions to warming from the terrestrial system seeing this enhanced CO2. And the red here is showing where you're getting warming, just even though the radiation in the atmosphere is seeing the pre-industrial CO2, we're getting warming just from the terrestrial system of the physiological aspect of it. And in the bottom panel, the fraction of the transient climate response, we see that we're up a non-trivial fraction of the model warming is just from this physiological response. And the take-home message from this is that is the first part of the text says what happens in the carbon cycle, particularly the terrestrial carbon cycle, doesn't stay there. It couples to the rest of the model. I'm running short on time, and so I'm going to actually skip this slide here.
You still have 7 minutes, Keith.
Oh, I thought, thank you for correcting me. I was looking at the wrong part of the time. Okay, thank you. This is some analysis of community projects of the DPLE where Nikki Lovendusky is looking at the carbon cycle response, the carbon cycle behavior in this community project of the decadal prediction large ensemble. So this is an experiment that was based off of the lens experiments, and then decadal prediction experiments were performed with that code base. And then Nikki is looking at the ocean carbon cycle in those simulations. The black line in these plots is a reconstruction of air-sea CO2 fluxes globally averaged. And this is from a hindcast experiment forced with reanalyses with the ocean ice system. So we don't have a great historical record of global air-sea CO2, observed air-sea CO2 fluxes going back into the mid-20th century. And so we're comparing the decadal prediction experiments to this reconstruction forced with reanalyses. And the one-year predictions, whether they are using the raw atmospheric CO2 or the detrended, shows a really good match between the reconstruction and the initialized forecast. And it's doing better than we do with the uninitialized forecast, which doesn't get the wiggles that are present in the reconstruction or the predictions that arise from interannual variability. When we do compare to the observational product, this Landshutzer neural net product, we do see that we're not capturing the full magnitude that is present in that product for estimates of the variability of the CO2 flux, and the understanding the mismatch between the initialized forecast and the Landshutzer observational product is a work in progress. This is really just trying to sample some different aspects of looking at the carbon cycle that's going on in the working group. Some additional activities that are going on. Some aspects of these activities impact everyone who's doing carbon cycle science in the coupled model. In particular, as you've probably heard in other parts of this workshop, the Ocean Model Working Group is transitioning from POP to MOM6 for CESM 3. And so the carbon cycle, ocean carbon cycle model is being ported from POP to MOM. And so what is happening is that our biogeochemistry library Marble is being coupled to MOM, taking the coupling that we currently have to POP. Mike Levy is the software engineer working on this, and he's accomplished a lot of the majority of the coupling of the state fields and exchanging information and diagnostics from the Marble BGC library into the framework that is present in MOM6. There are still some scientific issues that we are dealing with as we switch to MOM6. And an example that we're working out the details is that the vertical grid in MOM6 is time varying. And in the ocean interior, we expect we'll be using a something close to an isopycnal vertical coordinate. In the way that MOM implements this ends up leading to potentially having very thin layers at the seafloor, where you have densities in your coordinate system that are not present at a particular location on the globe.
Keith, we should probably move towards questions soon if you can wrap this
Okay.
up.
Thank you. And so there's some additional details there being worked out. There's a number of activities that are being worked on, including more functional groups in the ocean model, BGC, improved sinking particle dynamics with some new observational products, coupling to higher trophic levels. There's a fish project that was mentioned in the Paleo Working Group earlier today. And there's some additional projects that I don't need to read the text to you. When we get to the end of the session here, we're going to have some discussion on how to coordinate BGC activities. And I'm just going to leave you with sort of the motivation for this discussion section. We have a goal of reinvigorating activity within the Biogeochemistry Working Group. The number of presentations that have been submitted to the BGC Working Group has been down in recent years. And there's a number of reasons for that. Not all of them are necessarily bad. Some of that is migration of carbon cycle talks to other working groups where perhaps they have a better fit for other aspects of that. But we do want to make sure that we have a vigorous activity going on in the BGC group. And so we want to have some discussion about how to have a more lively working group. And so in terms of thinking about that, well, a natural question is, well, what is the purpose or goal of the BGC working group? And there's a few different ones that the working group co-chairs have mentioned, whether that's the support of community projects beyond the scope of other working groups, like the coupled carbon modeling effort that originated with the flying leap that Inez coined many years ago, running experiments that are community experiments that are analyzed by different members of the working group, such as the C4MIP experiments, or taking part in the experiments being run out beyond the typical 2100 out to 2300, or analyzing BGC aspects of other community projects, whether it's a LENS DPLE or CLM PPE. And so some questions we're going to try to have people have some answers when we have the breakout groups are, what parts of these aspects of BGC are you most excited about? What's your level of interest in these activities, or other things that you're interested in seeing the BGC working group? And what can the BGC working group do to support these activities? There is a mailing list for the working group that at this point is not heavily utilized, and that's a resource that we could certainly take more advantage of in the future. So I'll close there and we'll look at questions, take questions now.
Great, and we already have some questions in the chat. I don't know if Nikki wants to speak up, or I can read the question.
Can you hear me?
Yeah, go ahead.
Okay, so Keith, why is the ocean so sluggish in CESM2? And is it worse than it was before?
We have some theories about what's going on. There might be a salinity bias in the Southern Ocean, a fresh salinity bias that is reducing ventilation. We might have some problems with too restrictive topography. It does seem like it's worse than it was in CESM1. When we look at those radiocarbon ages, they do seem like we've degraded compared to what we previously in previous versions of the model. but we didn't catch it until too late in the development cycle.
So what does that mean then for like heat uptake and our transient climate simulations that are using this CESM2 ocean configuration?
That's a great question. I think it's all about time scales. And so for the BGC, I think this has really hit us when we do the spin up simulation, where we run for thousands of years to spin up the carbon cycle. And then that really becomes evident. For something like a centennial scale transient experiment, I wouldn't guess that it would be a super big impact, but I could be wrong. But my hunch is that we're seeing it in the carbon cycle because of the long spin up that we do. I see Will has a question.
Yeah, I can summarize from the chat. Will asked about the changes in temperature that Keith showed and whether those were caused by increases in water use efficiency and lower land latent heat flux. And Claire responded and said, yes, the plant responses to CO2 are leading to lower latent heat flux in those coupled simulations, which drives warming. And then Danica also asked if that increases as a reaccounting for changes in leaf area index. And I'll just add that that was a analysis of about 12 CMIP6 models. So those are the carbon cycle enabled models and they have varying leaf area, but those are the simulations that we were utilizing for that rather than running our own simulations. But yeah, that ET is due to stomatal closure, which is even though the leaf area is increasing, the stomatal closure still wins.
Right, we should, at time, so we should probably switch to the next talk. Thanks, Keith.
Great,
Sure.
And our next speaker is Danica Lombardozzi.
And I need to make sure I find the button to unshare, excuse me. So Danica, you should be able to share now.
Okay, so can everyone see that? Yes, great. And it seems like I must sound okay. Thumbs up. Great. So I'm going to talk today a little bit about seasonal amplitude of CO2 exchange in the CESM2 and the role of agriculture management. So this is just a continuation of work that I've been working on for quite some time. So some of you may have seen parts of this presentation before, but hopefully these new updates are interesting and exciting to everyone. So I just want to start by highlighting that, as you all know, carbon dioxide is plays a really important role in our climate and has been increasing through time. But what I want to highlight even more is this annual cycle in carbon dioxide. And the reason that I want to highlight that is because every year, the carbon dioxide concentration typically peaks in the Northern Hemisphere early spring. And then as the Northern Hemisphere growing season progresses, the plants start to take carbon dioxide out of the atmosphere, which reduces the carbon dioxide concentration. And so I guess the difference between the high and the low values each year is what I'm calling the seasonal cycle amplitude. And this is because this is driven by plant processes, it's largely a measure of terrestrial carbon dioxide uptake. So the changes in the amplitude can tell us something about terrestrial carbon fluxes. And in fact, there are observed increases in the amplitude of the seasonal carbon dioxide cycle. So that's illustrated here for Mauna Loa and for Barrow, and this is work by Graven and colleagues. And so what you see is that there are strong increases in seasonal amplitude, particularly at high latitudes like Barrow, but still even at lower latitudes like Mauna Loa, we are seeing increases in carbon dioxide amplitude. So what in fact contributes to the changes in carbon dioxide amplitude? There are a variety of factors that might contribute, but there's still a lack of consensus in the scientific community despite this continual investigation. And so there's some contribution from increased productivity in high latitudes, Some of this is driven by carbon dioxide fertilization and warmer temperatures. And there are also contributions from agriculture, but in fact, no study has been able to fully explain the changed carbon dioxide amplitude. So models can be used to help disentangle these drivers, but if you look at the CMIP-5 suite of models, of Earth system models, most of these models, none of these models, in fact, capture the trends in amplitude. And so this is illustrated here for Point Barrow in Alaska, where you can see that the observations have a larger amplitude than any of the CMIP-5 models. And this is similar to the results in Graven, by Graven and colleagues, who showed that models also don't capture the amplitude north of 45 degrees. So very similar results. But if we look into the newer version of most of these, or of some of these Earth system models that were submitted to the CMIP-6 suite of simulations, you'll see that models have improved. And so now the CMIP-6 models bracket the observed changes in carbon dioxide amplitude. So I'm going to dig in specifically to the community Earth system model and look at the differences between CESM1 and CESM2. So I'm going to start by looking at the CO2 seasonal cycle in each version. And so this is similar to what Keith showed. And I've shown these plots in the past as well, but basically what I want to highlight here is this is the CO2 amplitude. So the change in CO2 amplitude on the x-axis over the year, so starting in January and ending in December. And what I want to highlight here is that CESM2, which is the purple line, has a larger amplitude than CESM1, which is the orange line, both at Mauna Loa and at Barrow. And CESM2 more closely matches observations, which are shown in the black line. It's still not perfect. We still have some work to do, as Keith said. But the amplitude is improved in CESM2. And you can also see that the trends in the CO2 amplitude are also improved in CESM2. And so this, you can see this for Barrow is on the top. Again, CESM2 is in the purple line and the observations are in the black line. And you can see that the change in CO2 amplitude for CESM2 tracks the observations relatively well, whereas there was not a strong change in CO2 amplitude in CESM1. At Mauna Loa, CESM2 is an improvement, but not a real strong improvement, I should say, for observations. But there is some improvement, and so we are moving in the right directions. And so, as Keith said, many of these improvements in CO2 and the CO2 seasonal cycle are coming from the community land model, or the land portion of the CESM. And so I wanted to know if improvements could be in part due to agriculture. So there are many changes between CESM1 and CESM2, but one of the larger changes that impacts the carbon cycle is the new explicit representation of agriculture in CESM2. And so just to give you a highlight, in CESM1, we represented agriculture as generic crops, which function similarly to C3 grasses, whereas in CESM2, we actively manage crops. So there are explicit crop types that are planted, harvested, irrigated, and fertilized. So how does agricultural management contribute to the seasonality of carbon dioxide? I also want to highlight that I included a photo here of fertilization because one of the biggest changes that we make to agricultural systems is to relieve nitrogen limitation on productivity through the application of industrial fertilizer. So I'm going to switch from looking specifically at CESM2 simulations and switch focus to the community Earth system models land component, the community land model, or CLM. And so using land-only simulations means that the land is running using observationally derived data atmosphere, and we're not accounting for changes in circulation. So I'm going to isolate the impact of both agricultural management, which you see here on the bottom, including fertilization and irrigation management, but I'm also going to look at the impact of forcings because other previous work has shown that these forcings are also important contributions to the net carbon fluxes. And so I'll look specifically at carbon dioxide, increases in carbon dioxide, and also changes in climate. And I also want to highlight here that I'm looking, because these are land-only simulations, I'm looking at the net flux of carbon from the land surface back to the atmosphere, the net flux between those two. And this is called net biome production. So I'm using this as a proxy for changes in CO2 concentrations from the land. So I'm going to start by showing you or highlighting the forcings, and then I'll move into the management after that in a series of next figures. So on the y-axis here is the net biome productivity amplitude, and on the x-axis is the change through time from 1850 through 2010. And I also want to highlight that the results that I'm showing you are for the northern hemisphere, so everything north of 30 degrees north. And I'll start here by highlighting the CLM5 results, which is the purple that you see here. So the change in biome production amplitude over the course of the growing season with all the bells and whistles in CLM. So it includes carbon dioxide, climate change, irrigation, and fertilization. So next I want to highlight the impact of climate change. So you see climate change in this orangish colored line here. And I just wanted to highlight that we do see an impact from climate change, although it's not a large impact, but we do see it, and it starts to emerge later in the 20th century. And when we look at spatial distributions of this change, you'll notice that most of the impact of climate change happens in the higher northern latitudes. So most of the changes are isolated to these northern latitude regions where the temperatures are getting warmer. So there is increased growth and productivity due to the warmer temperatures. And I should highlight here that all of the colors in this blue range mean that there are increases in net biome production due to climate change. I want to contrast that by looking at carbon dioxide fertilization. What you'll notice from this figure is that the magnitude of changes in carbon dioxide fertilization due to carbon dioxide fertilization are very similar to that of climate change, but you'll notice that the changes are more spatially widespread. So they're distributed over larger spatial areas than the changes in climate. And so you see that CO2 fertilization is important throughout most of temperate and high latitude regions. And then just going back to this time series plot, you can see that in fact, integrated over this larger spatial area, globally carbon dioxide has a larger impact than what climate had. And that impact really starts to become evident in the 1980s moving forward. So both climate and carbon dioxide have important impacts, but the impact of carbon dioxide fertilization is globally larger and globally more widespread than that of climate change. So next I want to dive into looking at the impacts of fertilization and irrigation management. So first I'll start with irrigation. The yellow or gold colored line here is the change in NBP amplitude or carbon flux amplitude when we turn irrigation off in the model. So when you don't have irrigation on, and you'll notice that globally this is actually a really small impact. And that's due to the fact that there's only a small fraction of irrigated land area. But if you look at this regionally, you'll notice that it's quite important, especially in South Asia, where there are large impacts. It's increasing the net biome production amplitude with a larger magnitude than we saw in either CO2 or climate change fluxes. And so to me, this suggests that in the future, as we increase irrigated area, irrigation will become globally more important, but it is regionally very important right now. I want to contrast this again with agricultural nitrogen fertilization, and just to show that the magnitude of impact for agricultural nitrogen fertilization is similar to that of irrigation, but it is spatially much more widespread. So these changes are more widespread and also larger magnitude than that of irrigation, but also of CO2 fertilization and climate change. So agricultural nitrogen fertilization increases CO2 amplitude. And if you look back at this time series, you'll notice that globally, this fertilizer really does have the largest impact on carbon fluxes between the land surface and the atmosphere. And these changes are, so the fertilizer is a larger driver of carbon dioxide seasonality than any of the other drivers. And it's evident earlier on in the time series, starting in the 1950s, for example, when industrial nitrogen fertilizer application became more commonplace. So I just want to end by highlighting, this is the figure that I just showed you on the top with CLM, the change in net biome production amplitude in CLM in the land-only simulations. And when you turn off agricultural nitrogen fertilizer in CESM2, so in the coupled model, you can look at the change in the carbon dioxide amplitude and see that the patterns, the spatial patterns of change are actually similar to that of NVP, but you do see changes a little bit more distributed due to the fact that this CESM2 does include atmospheric circulation. And these changes in carbon dioxide amplitude are actually quite large. If you look at these agricultural areas, you're getting changes of up to 9 parts per million in the amplitude in some of these regions. So I just want to end by summarizing and really highlighting that agriculture does have an important impact on carbon dioxide fluxes. When you look at the difference between CESM2 and CESM1 and CESM2, you'll notice that the CO2 amplitude is larger in CESM2 than it is in CESM1. And agriculture is a large contributor to those changes in the carbon dioxide seasonal amplitude. And this is primarily due to the impact of industrial nitrogen fertilizer. So fertilizing our crops is a really important component of the terrestrial carbon cycle and changing those seasonal flux patterns. I don't want to say that nitrogen fertilization is the only impact because in fact, both carbon dioxide fertilization and climate change contribute to this increasing amplitude. So I will leave it there and I'm happy to answer questions.
Thanks. We've got about 5 minutes for questions.
Great, you can go ahead and type a question in the chat or raise your hand, or barring that, someone could speak up as well. Okay, go ahead, Stephanie.
Nice talk, Danica. So my question is regarding agriculture. There's also agricultural burning to clear land. Is this also included in these updates? And how would it affect the carbon cycle?
Yeah, there is agricultural burning included in CLM. So that is a part of these simulations. I didn't look specifically about how agricultural burning affects these carbon dioxide fluxes, but I imagine that it does. I think that we don't fully, we need a better representation of agricultural fires, actually, because we don't account for example, shifting cultivation or slash and burn fires. We just account for general agricultural fires. So it likely contributes, but I'm guessing that the impact of fire is underestimated in these simulations.
We'll go to Peter next, and then I'll come back to the questions in the chat.
So Danica, why is the irrigation signal so large in the Nepal region?
There's just a lot of irrigated area in that region. I think a large part of that region is agriculture land, and a large part of the agriculture land in that region is irrigated. And so that's part of the reason why I think it's about 25% of total land area is cropland, and then, or no, sorry, it's 10% of total land area is cropland. And then in that 10%, only 25% of cropland is irrigated. And so irrigated fraction is actually really small, which is why you don't see a lot of irrigation impacts elsewhere in the world. But that is one region where irrigation is nearly 100% of the cropland area. And there's a really high fraction of cropland in that region.
Great. A question from Genji Liu. Can CESM2 capture the greening trend over the high latitudes?
That's a great question. I have not looked specifically at that, but I do think that productivity increases in CESM2. I know it does within CLM. So but I would have to get back to you on that.
Yeah, it connects back to what Keith showed of the growth rate of CO2 and what the contributions might be from northern greening. And then Britt asks, does fertilizer have an impact on annual mean NBP and CO2 growth rate?
I think likely does have an impact on CO2 growth rate, but it's not something that I probed for sure. It's to know for sure. I would expect that it does impact annual mean MBP, but maybe not as largely as changing the amplitude, because we also are harvesting the crops every year. So that biomass is removed.
I guess I'll ask a question too, which is that you showed in that last plot a CESM spatial map of the impact of fertilization. And I was wondering if you've gone and calculated the amplitude specifically at some of the observational locations like Mauna Loa and Barrow to get from whatever simulations you have the attribution to these specific things you're looking at. It'd be really cool to see the attribution to all of them, but if you don't, I know that you can't quite run all of those experiments, but for whatever experiments you do have you had a chance to think about looking at that?
Yeah, so I've looked at it for fertilization specifically because that's the only one of these perturbation experiments that I have in the fully coupled model. And in looking at fertilizer, it does contribute. It's not all of the difference, but it does have an impact that is actually measurable. So.
I was specifically wondering if the attribution would be very different at Barrow and Mauna Loa, since you showed the different spatial patterns with the climate effects being more prominent at higher latitudes.
Yeah, I do. I have that figure somewhere. I don't remember actually what it looks like, but yeah, it's a good point to see how much it plays a role in the latitudinal distribution also of CO2.
Right, and that's about time. Thanks so much, Danica.
Yeah, thank you.
Okay, next up we have Will.
Hello, let's see. Oops. This is my first Zoom presentation. I got to figure out how to do it. All right, you guys seeing this?
We see your slide. Yep.
Yep. All right, good. Not the first one though. That's too bad. Good.
But they are changing, so that's
good. Great. I'm Will Weeder. I'm the co-chair of the Land Model Working Group, and I realized that I haven't actually given a science talk at a CESM meeting for a while. So thanks, Keith, for the invitation to present. This is building off the same set of simulations that Keith talked about and that Danica talked about, but specifically, I'm looking at interannual and seasonal drivers of carbon cycle variability represented by CESM2 ESM. And this is work that I've been doing with Keith and Danica, as well as Zach Butterfield and Gretchen Keppel-Alex, who are both, Zach's a former graduate student at University of Michigan. And it's a paper that is just about accepted in global biogeochemical cycles and will be in the CESM2 special issue, and was made possible by a NASA project that Gretchen and I had. So before I kind of dive into the problems that we diagnosed with the model, I want to highlight that CLM and CESM have an improved carbon cycle representation. So these are just two ILAM plots of ecosystem and carbon cycle metrics for the CLM5 GSWP3 runs that Dave published on in his 2019 James paper. And then for CESM2 compared to the lens, the kind of take home here is that CLM5 and CESM2 have a lot of green boxes, kind of no matter how you look at it, except for in soil carbon, which is, I guess, if the oceans have poor deep water ventilation, soil carbon still is the bane of CESM2. On the land side, that's not what I'm going to talk about here. And specifically, I want to focus on this carbon dioxide metric a little bit, which is, I like to call this the Gretchen metric, and this is kind of what it looks like in ILAM space. And so if you're not familiar at looking at ILAM plots, you can drill down from these kind of stoplight kind of color diagrams to look at individual metrics. And so this is what CO2 looks like. And for CLM4, CLM45, and CLM5 shown here, you can see that CLM5 scores better for all of these different metrics except for interannual variability. So this is kind of a known deficiency in CLM5, and I wanted to try to investigate that a little bit more. This is the kind of cool diagnostic that Gretchen came up with and what it looks like. This is what NOAA observations suggest the interannual variability of CO2 in the atmosphere is, roughly 0.6 to 1 ppm, depending on where you are latitudinally. And both CLM4 and CLM5 underestimate that by quite a bit. So the two kind of things I want to explore here are first, what's causing this low carbon cycle variability in CESM2. And then also I wanted to look at seasonal drivers of carbon cycle variability. So it's a little bit of a whirlwind to try to squish these two things into a relatively short talk. And just kind of to ground everyone with what I'm talking about. So I'm using one ensemble member of the CESM2 historical simulations. All of the results are from monthly detrended anomalies, and I'm focusing here on ecosystem fluxes, so NEP, net ecosystem production, which is just the difference between GPP and ecosystem respiration, as well as climate drivers, so temperature and water storage, TBOT and TWS. And I'm just focusing on kind of the GCP era, so 1960 to 2014, the end of the historical simulations. So there's a lot here, and I'll kind of walk through all these panels. I realized I don't know if I have a, can you guys see this? If I just give me a marker that you can see.
We can see your cursor, just a little bit.
Right.
Yeah.
I wonder, that's all right. That's good enough for now. So in the top left panel, what's shown in green is the GCP atmospheric growth rate detrended. And it's been reversed for convention so that positive fluxes are net land uptake and negative fluxes are land losses. And then the interannual variability of CESM2 is shown in black. And you wouldn't expect CESM2 to match that, the exact wiggles of the observed, because we don't have El Niños and La Niñas in the same time, although we do have volcanic eruptions, so effects of Pinatubo on the carbon cycle and other volcanic eruptions should be evident, although they're pretty weak in these simulations. But the take home is that the magnitude of the wiggles, the standard deviation of the flux anomalies, is roughly half what it should be. So it's roughly 0.5 petagrams carbon per year is the standard deviation of the NEP anomalies. And from the Global Carbon Cycle Project, it looks like they should be closer to 1. So because of that low carbon cycle variability, we end up seeing relatively low carbon cycle sensitivity. Here, I'm just plotting NEP anomalies against terrestrial water storage and tropical temperature anomalies. So this is supposed to be a comparison to the Humphreys et al. Paper from 2018. And then for temperature, the Cox et al. 2013 paper. There's a few that have looked at temperature variability. And you can see for both of these metrics, because the magnitude of the y-axis is compressed, the slope of the line is too shallow. The other interesting part is that even though this is with a fully coupled model, and so we have modeled terrestrial water storage and temperature anomalies, the magnitude of the x-axes here are kind of spot on with the observations. So it looks like CESM is doing a relatively good job representing the climate variability, at least over the end of the 20th century and start of the 21st century, but we're not doing a good job capturing the vertical component. Again, that's kind of a known deficiency, but this is what it looks like in terms of the sensitivity of net ecosystem production to water and temperature anomalies. So what could be causing that? Again, NEP is just the difference between GPP and ecosystem respiration. So a low net flux, low variability in the net flux could be caused by low component variability. So low variability in GPP, or it could also be caused by this high component correlation. So if GPP and ecosystem respiration co-vary at kind of the same time scales, then the net flux will necessarily have a low interannual variability. And so there's some preliminary results from Matt Wozniak, who is another one of Gretchen's students, looking at GPP fluxes this is from the UMBS Flux Tower site. And you can see that the default parameterization for this version of CLM has a pretty low cap in GPP and very narrow kind of ranges. They thought this might be because of the parameterization of diffuse versus direct sunlight. And so they changed that parameterization here and got a little bit higher cap and a little bit greater variability. But the take home, or at least for my purposes here, is that CLM doesn't capture the full range of variability in the observations, even for this Flux Tower simulation where we're giving it observed meteorology. So we don't capture these kind of hot moments of high productivity, and we don't have the same magnitude of range of GPP. So at least at this one Flux Tower in northern Michigan, there's some indication that the inputs here, GPP, are potentially too low compared to the observations. You can look at that spatially now. So these are from the CESM results, and these are the standard deviation of detrended anomalies. And to my eye, what kind of jumps out is that there is relatively high standard deviation of the anomalies in these arid savanna regions, which is in good agreement with what a lot of folks have seen, that these arid regions are potentially drivers of carbon cycle, have high importance in terms of carbon cycle variability. But if you look in the core of the tropics, it's really, really low. And if you normalize for the fluxes, the normalized standard deviation, or the coefficient of variation is incredibly low. And that's a feature that's common in many CMET models. So this O'Sullivan paper really called it out for the trendy models. Many land models have this kind of chronically low bias in tropical forests, which is one potential reason that CESM2 has low carbon cycle variability. So the other potential, this low GPP variability is one potential driver. The other is a high correlation between GPP and ecosystem respiration anomalies. So this is a plot of what that looks like, and this correlation is really, really high. Some work that Dennis Baldacci did at FluxNet, it's kind of a collection of FluxNet sites, suggests that the slope of this line and the correlation should be much lower than what we're getting in CLM or CESM in this case. And so I wanted to dive a little bit into trying to diagnose this. And so this is now scatter plots looking at ecosystem respiration anomalies, and ecosystem respiration has two components. So there's an autotrophic respiration component shown in green and a heterotrophic respiration component shown in blue. And you can see that the anomalies in ecosystem respiration are largely driven by the autotrophic component. So then drilling down one layer further, there's three components of autotrophic respiration that are related to growth respiration, maintenance respiration, and fun. And so fun is this new feature that's in CLM5 that makes plants pay the carbon cost of nitrogen uptake. But the way that it's applied, it's a relatively large flux. And you can see here that it's tightly correlated with the autotrophic respiration anomalies. And so, most of the variability in autotrophic respiration is instantaneous, and it's related to this fun flux. And so, although it's not we can't really turn fun off in CLM5 to say that it's fun that's causing this. But my guess is that because of the way that we've kind of put CLM5 together, that this high correlation between fun fluxes and GPP are what's driving this high correlation between GPP and ecosystem respiration. Okay, so switching gears, stop thinking about variability. To me, that's like the useful part of the talk. This is the fun part of the talk. Thinking about seasonal drivers of carbon cycle variability. So now I'm going to start looking at monthly anomalies instead of these annual anomalies. And what I wanted to do was use some of the work that Gretchen's lab had done looking at singular value decomposition. This was some new math to me. It basically works similarly to an EOF, if you're more familiar with that. And what happens is you decompose a matrix of interannual variability that's dimensioned month by year. So in this case, 12 months by 55 years. And what comes out of it is this component that I'm going to be calling the vectors, or the singular value vectors, and then the weights. And so we'll focus here first on the vectors that come out of the SVD, and focus on the first two vectors from the SVD analysis. And then second, I'll go back and look at the weights. Sorry. And so this is what it looks like in kind of more intuitive space. And so what's shown in this figure in gray is just the climatology of GPP. This is for the kind of high latitude ecosystems north of 50 degrees. And the units on the y-axis are kind of meaningless for this type of activity. And then time, so month of year, is shown on the x-axis. So the climatology of GPP in northern latitudes has this really, kind of predictable green up in the summer and then senescence in the fall. And so what I'm calling the amplification vector is strongly correlated with that climatology of GPP. So I identified the amplification vector as the vector of the first two that most strongly correlates with the climatology of GPP. The other feature that comes out of the SVD analysis is this theta estimation. And so for positive values, basically you can think of it means that the anomalies are above the 0 line. And so a theta value closer to 1 means that you've got an increase in the amplification of GPP. And for these grid cells, it's 56% of the variation in GPP can be attributed to, looks like this amplification vector. The second most common mode of variability is what I'm calling the redistribution vector. And so the redistribution vector is characterized by this kind of early spring green up, and then subsequently a summer and fall decline in GPP. So this kind of positive anomaly in the spring, followed by a negative anomaly in the late summer and fall. As a result of having kind of balanced, positive and negative phases, the theta value is closer to 0. And for these grid cells, roughly 26% of the variation can be attributed to years that have this kind of characteristic pattern in them. So moving forward, I'll kind of go through characteristics of the amplification vector and redistribution vector, but I'll kind of keep referring to this figure because I find it useful to understand what's going on.
You've got 5 minutes, Will.
Perfect. So here is what this amplification and redistribution vectors look like. Roughly 45% of the variance globally can be attributed to this amplification vector, and 29% of the variance can be attributed to the redistribution vector. But there are some areas, especially in kind of the Eastern United States, much of Central Europe, that are really dominated by this redistribution vector, which is something I'll come back to. You can look at it in latitudinal bands. So again, we've seen this one before. It's kind of characteristic. It's easier to see at mid-latitudes, high and mid-latitudes, but even the tropics shown here for the Northern Hemisphere and the Southern Hemisphere show these kind of characteristic amplification and redistribution vectors. So these two modes of variability seem pretty pervasive. And what I wanted to do then was correlate the weights that come out of the SVD analysis with seasonal anomalies. So here I'm looking at seasonal anomalies of GPP in winter, spring, Northern Hemisphere, winter, spring, summer, and fall. And kind of by definition, this amplification vector is characterized by positive correlations with GPP in the summer, especially in the Northern Hemisphere. So that's what's shown here is a high correlation between the SVD weights and the JJA seasonal anomalies of GPP. So this is just kind of a sniff test to make sure things are working right. And then I wanted to look at anomalies, how that correlates with terrestrial water storage and temperature anomalies. And so there's a lot to look at here. I'll just draw your eye to this kind of the summer panel. And what you see is that the amplification vector is largely driven by water. So positive correlations with water anomalies in mid and low latitudes, and then also positive correlations with temperature in the high latitudes. So not surprising if you warm up the Arctic, you do see these positive amplification vectors. It gets more interesting for redistribution. And so redistribution is characterized again by this kind of early summer green up, which in the Northern Hemisphere is most strongly correlated with warmer springs. So that's interesting. You get, a warm up in the spring, leaves come on, and you get this positive anomaly. But then the negative anomaly is shown here, and you get kind of negative anomalies that tend to be correlated again, possibly with heat stress, but also increasingly with water storage and TWS anomalies in both the summer and fall. So water stress in the summer is associated with these negative GPP anomalies.
Well, you've got about 2 minutes left.
Okay, I'm just going to cut the summary and get to I think what the cool future things to do with this could be. I think it'd be awesome to look at this in future scenarios. So this is just kind of for the historical record, and it's ripe to do this with a large ensemble. It also points to some opportunities of doing ecosystem predictability work, and I think SMILE would be an opportunity to do that with. And then looking at other models and observations would be useful. This is not work that I have a lot of time to do. So if anyone's interested in doing it, I would be more than happy to share the code that's already on GitHub and talk more about it. That's it.
Could you just define SMILE quickly?
Oh, sure. SMILE is I don't even remember what the acronym stands for, but it's part of the Earth System Prediction Working Group, and there's these series of 2-year-long initialized ensembles that Steve Yeager is running.
Seasonal to multi-year large ensemble. Thanks, Nikki.
Hey. Thanks.
Jinji had a question. Are these variables, water storage and temperature at the bottom, from observations or from CESM2?
That's from CESM2.
So the self-consistent climate that the GPP was experiencing in the model.
Right. That's right. Yep. Mm-hmm. I did the same analysis with CLM5 offline, and you see pretty similar patterns.
Other questions, you're welcome to type in the chat or raise your hand. Or here, somebody might have unmuted.
I think that was me, just to say we probably only have time for one question, if that.
I guess I would say, what's your hope for, do you have any idea of, do you think the interannual variability problem is likely to be improved? Do you have any ideas about how one would improve that?
I think the fun fluxes are pretty large, especially in future scenarios. And so I think it kind of, I'm hoping that with the parameter perturbation experiment that Dave and others are doing, that we get to kind of explore ways that we can redistribute the autotrophic respiration fluxes maybe away from fun into some of the other components.
Oh yeah, that's really interesting to think about how the PPE might help identify places in the parameter space where there is more interannual variability.
Yep. Yeah, I think that'll be a neat opportunity with that work.
Great. Okay, I'm guessing we
Thanks,
need
everyone.
to move on. Thank you. Next we have Ming Yang.
Is it showing all right?
I see so far not in presenter mode, but I see your screen. There you go.
Okay.
Go ahead.
Thank you. So my name is Ming Yang Guo. I'm in my final year of PhD at Purdue University. So people have been talking about CO2 a lot. I just kind of want to bring some attention to methane also. I work on lakes, including all the lakes, ponds, and reservoirs in this case, because they are very important methane sources. Actually, the second largest natural source after wetlands. The emissions are quite disproportionate compared to the surface area covered by them. Here is a schematic diagram showing lake methane processes. So methane is produced in the anoxic lake sediment by methanogens. And there are 3 pathways to transport them to the atmosphere. There is mediation by aquatic plants and diffusion, on the way of which methane will get oxidized in the water column by methanotrophs. And then there's also ebullition, which is just bubbling so that methane can be protected from oxidation. So that's why Evolution can be the dominant pathway in many lakes. Diffusion and evolution are the most studied pathways, by the way. Also, another important reason is that lakes are especially abundant in the northern high latitudes. About 40% of the world's lake area is north of 50° north, meaning that they'll probably undergo great impacts by climate change. And speaking of climate impacts, there are many ways that this can work. Most directly, warming will enhance microbial activities, while methanogens are found to be more responsive to warming than methanotrophs, meaning that methane production will increase faster than oxidation. And for lakes that freeze in winter, they'll have longer ice-free days, meaning longer emission periods. Also, there are impacts related to permafrost thawing. And except from that, climate warming can also work with other factors such as lake eutrophication, meaning that they're getting more nutrient rich. Lake eutrophication will exacerbate the warming impacts. I'm just making several examples here, and this list can go on, and they're all potentially very important topics. So given all the necessity to study lake methane emission, our estimations so far still have large uncertainties, actually the largest among all natural sources. So these two data synthesis studies show the gap between top-down and bottom-up estimations. Top-down here means satellite data inversion, and bottom-up means modeling or statistical extrapolation. So in this more recent paper in 2020 by Sono et al., the freshwater emission by bottom-up, as shown here, is around 150 teragram per year. But if you look at the top-down estimation, it's only less than 40 for all natural sources except for wetlands. So that's like 5 times. So the bottom-up estimation is like 5 times of the total of the top-down estimation. And if you compare it to the result in 2013 paper, this gap really hasn't been reduced, but rather enlarged a lot. This can be due to various reasons with bottom-up estimations. Well, the top-down method has its own issues, but it's not within the scope of my talk. So the bottom-up estimations rely on field observation data. Well, most data are for diffusive fluxes only because they're easier to measure. But as I mentioned, evolution can be the dominant pathway. And also most observations only lasted for less than 3 days because of the measurement difficulties, which is far from enough to capture the lake's annual emission potential. And there are many measurements for very small lakes, which are, of course, highly productive and and the results can be sensational. But when it comes to regional or global estimations, the mapping of these very small lakes can be a large uncertainty source. I'll talk a little more on that later. Also, the tropics is often underrepresented, although it has larger emission than the Arctics, which receives much more attention. And in terms of the methods, an intrinsic issue with statistical extrapolation is that it's not spatially or temporally explicit. And for example, for northern lakes, it uses an estimated ice-free length for all lakes or by latitude. Either way, it causes large uncertainties. And for modeling, since there are more than 1 million lakes over the world, the process can be very time-consuming. Also, it's hard to calibrate the model to suit global lakes because model calibration can be really tricky sometimes, especially when we're looking at a global scale. And of course, both methods can be biased by the data availability if not taken carefully. So with this in mind, the goal of this study is using a modeling approach to reduce the uncertainties in the estimation of global lake methane emissions and to make projections of future emissions under different climate scenarios. And also to investigate the spatial distribution, which is a wonderful thing of model. I'll briefly introduce the model I'm using, specifically how it compares to CLM, the lake model in CLM, and then go through the simulation design quickly because the modeling details are not what we want to get into here. So the model I'm using is called the Arctic Lake Biogeochemical Model, ALBM. It was originally built for the Arctic, but has been modified and validated on lakes in other regions. It's a participant model in the ESME project, the intersectoral model intercomparison project. So it has been applied in several studies and also model intercomparison work. The two models share many similarities. They are both 1D process-based, and the thermal modules are quite similar. They're both based on the Haus-Tedler model, meaning that they simulate key processes like turbulent heat fluxes, ID diffusivity, and thermal conductivity and radiative frothing in very similar ways. Basically, the algorithms are interchangeable. But when it comes down to what exact algorithms are used, there are differences. In fact, we did an algorithm-based model intercomparison study and compared in total 18 algorithms of key thermal processes and selected one with the best performances for regional or larger scale simulations. The paper has just been accepted. And other from that, in terms of the methane model, CLM only has, well, the methane model in CLM is mainly aimed for wetlands, which are in some ways similar to lakes, but also have many different mechanisms from lakes. For example, the gas transportation are quite different, and also CLM prescribe bulk oxygen concentrations to the grids instead of simulating them explicitly. So for model calibration and validation, we carefully filtered the data and selected those, like for northern lakes, only those with at least 3 months observation during at least one as-off season. And for tropical lakes, the data have to cover both the wet and dry season for at least 1 year. That left us with 84 lakes, 39 with evaluation measurements. So we calibrated and validated the model within each subgroup. And the same way we did the global simulation, we used, for the general lake mapping, we used hydro lakes, which includes all lakes larger than 0.1 square kilometer. And for lake classification, there are all kinds of data set we used. There was a lot of data organization work. And finally, we ran simulation for the first decade and the last decade of the century and considered 2 climate scenarios, the RCP 4.5 and 8.5. And here are the results. For the present, we simulate a mean annual emission of 25.8 plus minus 9.4 teragram. The tropics, as shown here, is the largest source, even though it only has about 1/10 of the lakes in the subarctic region. And by the end of the century, the total emission is estimated to increase by 24.2% and 45.3% under RCP 4.5 and 8.5, respectively. The temperate zone will become the largest source under RCP 4.5, and under RCP 8.5, the Arctic and subarctic region will also exceed the tropics. Comparing to the previous studies, our estimation of present emission is on the lower end of these ranges. So for this Rose and Trader paper published very recently, they did include lakes smaller than even 0.001 square kilometer, but I only truncated their results here just for comparison. Our result is in some way more reasonable to reconcile with the top-down estimation. Well, there are definitely limitations there, and I'll go to that shortly. To investigate the sensitivities to climate, we calculate the percentage increase, which is a delta E CH4 here, and also the delta E CH4 over the change in the mean summer air temperature. So it shows that the subarctic and Arctic region will experience the severest increase in methane emission, around 60% higher than the temperate and almost more than triple the tropics. And this region is also the most sensitive to the climate intuitively and from the simulations, with more than 10% increase with 1 degree warming, so that more than double the tropics. Well, despite the different climate impacts on different regions, the spatial distribution, on the contrary, is not changing much. So here are the global mapping and the latitudinal distribution of methane fluxes. And there are scenarios, the mean annual methane flux peaks at around 10 degrees north and decreases towards the poles. And the hotspots on the maps are more or less the same. So here is a summary of our current results. We estimate and predict global lake methane emissions for the first and last decade of the century. We found that the tropics is the largest source at present, but the temperate and Arctic and subarctic Arctic regions will become more important in the future. Especially the Arctic and subarctic is the fastest growing, as well as most climate sensitive, and therefore should receive more attention in the future projections. And probably more studies should be done on the mechanisms of Arctic lakes responding to climate. And finally, the spatial patterns within this century do not seem to be largely affected. So our studies has many limitations, and this small lake issue kind of comes to the fore throughout my talk. So here's the thing with it. The smallest lake in the dataset we're using is 0.1 square kilometer. There's one other spatially explicit dataset called global water bodies, or GLOWABLE, including lakes as small as 0.002 square kilometer. But to resolve these lakes on satellite images can lead to pretty large uncertainties. Also, that dataset does not include lake depth information. So we still went to hydro lakes for our purpose. And in terms of the statistical extrapolation studies mentioned previously, they included even smaller lakes. And the total surface area and abundance of these lakes obviously cannot be resolved on satellite images. So the relationship is obtained using a Pareto distribution as shown here. So basically, the abundance increases fast as the lake surface area decreases. However, some studies have found that these points can deviate away from the distribution when it comes to lake smaller than 0.5 square kilometer. And the discrepancy can even be an order of magnitude. So considering that in the statistical extrapolation studies, they estimate emissions from very small lakes as 40% of the total emission, it's really hard to estimate the uncertainties in the results.
You've got about 5 minutes left.
Okay, thank you. I'm about to wrap up. And especially when they'll be experiencing, especially when small lakes are abundant in the northern latitudes, meaning that they'll experience rapid lake dynamics of like expansion and drainage, rapid in terms of normal lake lifespan. So it really adds more and more to the overall uncertainties. And apart from that, the classification of global lakes can be limited by data availability. For example, like all lakes without trophic status data are considered oligotrophic, meaning that they're nutrient poor and thus less productive. this means that our estimation is potentially conservative. And while there are, of course, other limitations, and we're always trying to improve our study and experiment design. This is my last slide. So for future work, we want to further investigate like methane emission under climate change. We plan to like other impact factors due to climate change. For example, the permafrost thawing has been shown to be significant. They will increase the sediment carbon content due to the input of old organic carbon. And also permafrost affected lakes will experience drainage or expansion relatively rapidly. So that will affect the total emission largely also. And also, as mentioned, lake eutrophication can be another potential topic. So these all require more data collection and modeling. And yeah, that's the last slide of my talk. And thank you all for listening.
Great, thank you. We can, I think we probably have time for 1 or 2 questions.
1 or 2.
Yeah, so if you have a question, you can go ahead and put it in the chat or you can raise your hand. And I actually have a question. You said that your prediction for the future or projection for the future is that the spatial pattern of emissions wouldn't change very much, but you also mentioned that you expected the tropics to become relatively less important, or maybe the emissions to become relatively higher at high latitudes. And so I was just wondering if you could reconcile those 2 statements, because it seems like if you have a shift from one region to another, you'd get a shift in the spatial pattern. So could you explain a little bit more about that?
Right. I'll go to this one. So when I mentioned that the Arctics will become more important, I meant the total emission, like adding up over the whole region. But in terms of the spatial distribution, if you look at latitudinal distribution curves, the flux, which is in the unit of millimole per square meter, the pattern of that will still stays more or less unchanged. So meaning that for tropical lakes, they will still have larger emission per square meter. But if you're adding up the emission for the whole region, they're becoming less important than the Arctic. Does it make sense to you?
I'm still a little confused, but let's move on. It looks like Dave has a question. Go ahead, Dave.
Yep. My question is trying to understand how you initialize the model. Like where does the organic matter come from to generate the methane emissions? Is that a prognostic quantity or is it a prescribed quantity?
Yeah, that's prescribed. We collected data from previous studies, measurements on lake sediment organic carbon content, and like we assigned different organic carbon content to lakes in different regions. Like for Yadoma lakes, there was a value, and for thermokarst lakes, there was a value, and in that fashion, yeah, we use a prescribed value for each lake type.
So if you were going to try to do, I mean, if this was going to try to ever go into a fully coupled model, maybe you would have to figure out how to model that, or maybe just initialize it. It's been there for a long time, and I guess in most cases. So. Yeah,
that's a very good question, because the model can do that. There are those processes like the carbon burial and the carbon dynamics in the model, but well, this comes to the parameter issue, because I'm just not sure we have enough data to bracket these parameter values involved in those processes. So I just decided that probably that's a problem for future. We'll just use the prescribed values for now.
Yeah, makes sense.
Yeah,
Great, thank you so much. I think it's time to move on to our next talk.
thank you.
Our next speaker will be Xiaofeng. We
Yes.
have to stop sharing screen first, and then we can
Yeah,
share screen
I
again.
know.
No problem, no problem.
I'm sorry for that.
It's okay.
A little bit. Could you like force me to stop? Oh, I see the button. Sorry.
Okay, you're good now. And there we see Xiaofeng's screen. Excellent. Okay, go ahead.
Okay, guys, really glad to present some result from the model we really developed. It's not really developed, it's more like we improve a little bit based on CM model. So this model we call CM microbial. The name is really emphasize the microbial roles on the biochemical processes. So particularly in the current version, we emphasize on the soil carbon turnover and also on the methane dynamic. So that's why you see the title here is simulating the microbial mechanism for soil carbon and methane dynamics with the CM micro model. So I really want to emphasize, this work is, I did the early work for the model coding many things, but the latest many part of the presentation I show here has been done by my two students, Li Yuan and Wei. So you know the name here. Okay, so let's start with the real content. So this is the one of the report came out about 2 years, about 10 years ago, is 2011. So this is really a report by the American Academy of the Microbiology. So emphasize the incorporating microbial process into the climate model. So then after that, number model has been developed. One of these you see here is Steve Allison, Mimic, and Wader, and Corbus Mend resolves many different models. And also one of the models about really published by Josh is about 2003. So Josh always mentioned that it's a toy version model, but it more like the first, I think the very explicitly represent the microbial processes. That is one. So what I present here is we call CM micro. So you see the name. So that is really, so you see this is the structure. So all those, what I present here is the biochemical part, because this model is really built on the default the CM 4.5. So all the hydrology, thermal, plant are exactly same. So with the CM 4.5. So what we did is really based on the biochemical part, we add two parts. One is the biochemical reaction. So we add bacteria, fungi, and dissolved organic matter. And also we also add another called the microbial function group based method model. So those two parts is really the part what I will present in the following slide. So this is the really summarize the component we have been revised or some revisions we have done on the 4.5. So the bacterial part, bacteria fungi, so we separate two fungal groups, and also dissolved organic matter. This is for the biochemical. And also the method model I want to emphasize is we did have a very kind of like explicit variable here. So it include the dissolved organic matter. So acid, hydrogen acid classic methanegenesis, hydrogen trophic methanegenesis, anaerobic methane and oxidation, and anaerobic methane oxidation. And for each layer, so we have all those processes. And also for the three pathways for the methane transport, include the plant diffusion, plant media transport, diffusion, evolution. And also another things revision we have done is, so because current column based is sometimes cannot work well. So we reorganize the biochemical a little bit. So we use the predominant PFT type to parameter some microbial soil processes. In that way, for example, the defolding factor. So this is, we really parameterize to make sure that is each plant function type, it has individual value. So in that way, we can much better to simulate the soil carbon distributions. And also we bring some soil parameters and all the microbial parameters out into the parameter files. And the second revision is we allowing the vertical movement of the dissolved organic matter as the acid, methane, and the CO2 gas, right? So this is other than the perturbation in the default version. So we added even more function to allow those can move vertically, so around the 10 layers right now. So in the next two slides, I don't want to emphasize a lot. It really shows I showed a little bit what we do, the approach we call the microecological approach to improve the model. So for these two slides, I don't want to say a lot. Really, this is just show the approach we use. So one thing I have to emphasize, we in this approach and also in the later approach, we developed microbial mechanism. We try to put that mechanism in the CM. We include disposal, environmental filtering, microbial response, diversification, and local extinction. So those is the mechanism we trying to further improve the CM microbes models. So then in the next few slides, I try to introduce from two parts. So one is the biogeochemistry, the more like the carbon soil carbon dynamic, and second one is the methane dynamic. So this is the first part. So first part here is we really emphasize how the bacteria and fungi really regulate the carbon flow. So you guys know that in the default CM versions, we have all those cascade, right? From each pools, we have upstream. So I think this is Charlie Coven plot this figure very well. It really show that how this flow from upstream to the downstream. So for each individual flow from upstream to downstream, we added bacterial control and the fungal control. And for each of those, we have different CM ratio, and then they have different physiology properties, right? Because they have different physiology, so then when this carbon flow through the bacteria and fungi, so you have the CO2 release, then you can simulate how this microbial functional community structure change has impact on these flows. And also for the dissolved organic matter, so put here is really show that how the bacteria fungi can use dissolved organic matter, and then when it respires, it releases the CO2 back. So this is the general structure for that. So with that, so I think start 2 or 3 years ago, the UN has started trying to use this model, and we collect a lot of data from different sites across globe. We choose non-natural biomes. For each biome, we select more than two sites. Those is the site name. So every detailed information you can find in these papers. So all those different sites, we have one site for the calibration and one site for the validation. So all those represent individual site, and on the left, so this column left side is for the model simulate fungal biomass. On the right side is model simulate bacterial biomass. So for each site, I do want to allow you guys more like know that is you see here. So the different color of that represent, so that we change the scale a little bit to match the value, but the scale of the x-axis is different. Because the key reason for that is because the microbial diameter normally is larger compared with the ecosystem variable. So we try to keep this to make a comparison, but you see the value are different. So this is, I think this is urgent issue for the community if we are able to better simulate the microbial variables. So this is the distribution. Then this is the scatter plot to show that if we put all those for each biome, so you see roughly, so is the comparison here. So for the model can explain about 70% of the fungal biomass and about 26% of the bacterial biomass on average. So this is not very good, but roughly we are able to reach this is the current accuracy for the model. So after that, after we have used the model to simulate the bacterial and fungal dynamic, then we further look like, so how can we use this model to simulate the carbon processes? So in the next kind of few slides, so we really emphasize model simulate the microbial respiration. So all those different setups out. So you see, so each panel here represents as one site. So the red star represents the observation data. So the black dot is the model simulate. So for many sites, so the name of the site represents different site and biomes you see on the bottom. So this is the microbial respiration. So then this is the, we also further evaluate how the microbial seasonal variations can have impact on the different carbon fluxes. So this is the respiration. So bacteria, this is the biomass of the bacteria. So I want to let you guys know that is here. So the different color of that is we use two version models. So one version model, so we use the default CMX model, and this red color represented model, we remove the seasonal variation of the microbial control, particularly on the temperature and moisture. So we use annual average of the temperature, moisture, and to control the microbial biomass grow and dies, and everything else is same. So this is, in this way, we compare these two version model, then we can further evaluate how the microbial seasonal variations has impact on the carbon emission. So this is biomass. You see, so most of the biomes, so the seasonal variation did have a smaller, you see a smaller biomass of the bacteria and fungal. So this is respiration, and this is the carbon loss from bacterial fungi. So this loss eventually represent the microbial respiration, right? Because in the current version, all carbon release from soil to atmosphere, it must be of the bacteria and fungi. So this is the, so the red is the without seasonal variation, black is the with seasonal variation. So you see the different, this carbon loss, and this is carbon gain for the fungal and bacteria, right? So you see the different, due to the time limitation, I don't want to go detail, but those are the results we show here. Then we further to estimate, so if we add everything together, so how about the microbial respiration? And then what we got is this one, microbial respiration and carbon storage. So then really is, if you compare this, so the original model and model without season variation, so we did find that when we remove the seasonality, the respiration is decline, and then so organic carbon increase. In this we compare, we found that the microbial seasonality overall is stimulate microbial activity and then lead to higher respiration and the lower, little bit lower organic turnover. So this is the finding of this study. So this is some preliminary result we are currently working on, try to use this model to simulate global distribution of the different variables. So DOC, so organic carbon, microbial respiration. So this is the along the latitude distribution, and the right panel show the spatial distribution of the different variables. So bacteria, fungi, and the dissolved organic matters distributions. Okay, so I have a little bit time I want to introduce the method model and some application result. So this is the model structure. So similar with the previous version, but we add a little bit more content here. It's how can we use the current version to link with different field environment, so profile these different variables DOC as the acid and the CO2, methane, oxygen, and also how can we use this to connect with the genetic information. So some of has been done, some is still ongoing work. Okay, so this is the work done by another student, Yi Hui. So she worked on this model and use this model to simulate the methane fluxes and net oxygen exchange and also oxygen respiration at Arctic for the different landscape types. So this is for trough. This is for the low center polygon center. This is the low center polygon rims. So for the different landscape, we did have this model, the simulation of the methane fluxes. So this is for the either we have the sand tabs. So this is compared with observation. So then we further use this to simulate different tabs, right? You see, so we do have different values, different patterns, seasonal pattern of the different landscape tabs. This has been published 2 years ago after we done this one. So we further simulate on the site level, can we do the upscaling to see how this plot level measurement by the standard chamber, how can we upscale to the eddy Commons tower domains. So this work is just done about 1 month ago. So then this is the result for this model result. So we use the model, right? Using the CM micro model, and also we combine this model with the 3 type of the footprint model. So 3 type of the footprint model is these 3. So first one, the homogeneous footprint model, and gradient footprint model, and the dynamic footprint model. So the difference
5
between
minutes left.
Okay, thanks. So the difference between 3 footprint models, the homogeneous model will assume, so in a circle distant from the tower, every grid is have exact same contribution. So that's why we call homogeneous. The gradient footprint models, we have the site, the tower in the center, but the distance, the contribution to the tower measurement is inverse to the distance. So if we move away from the tower, the contribution is slower. So this is the contribution for the different grid to the eddy Commons measure the fluxes. And then dynamic is we use the dynamic footprint model to simulate each month. So how different site has different contribution to the tower measure the meson fluxes. So then this is for one site, but then we have 5 sites. So in the optic, the 5 sites. So, and also for the different years, different months, because some months we don't have data. So that's why you only see few months. And particular of those months is in the summer or in the fall season. But in the winter season, many of those need to have data available. So we use those to compare with observations. So roughly, so we can see that the homogeneous and gradient are dynamic. Most of those months, the dynamic model combined with the CM micro model, we have much better performance to simulate the methane dynamic. Even some months, we don't have the dynamic model is not best, but still it performed much better, right? So you see the R square here is still very high. So those and many other sites, for example, like the May of 2015, so the dynamic model is much better compared with other tools. So anyway, so this is the key conclusion for this is we are dynamic footprint model plus the CM micro model. We are able to much better to simulate the methane fluxes at the eddy Commons domain, tau domains. Okay, so I think that's all the content. Some ongoing workers, we are still working on this, trying to get more data to further improve model and also test model for different microbial mechanism, and also further to evaluate how the microbial contribute to the land estimate feedback, particularly on the CO2 and methane. And for nitric oxide, we are still working on it, but currently it's particularly on the methane and the CO2 fluxes. So I also want to acknowledge the contribution from many people over the past few years. So many colleagues or students has contributed to the data, contributed model, also contribute collect data to use data to validate model. Okay, so that's all. Thanks.
Great, thank you. Gretchen, can you give us a time update?
Yep, there's about 2.5 minutes left.
Okay, great. Do you have any questions? You're welcome to type in the chat or raise your hand. Go ahead, Will.
Well, it's good to see this work moving forward. I'm curious what controls the relative abundance of fungi and bacteria in your model.
Thanks, Will. So this in this current version, so with the bacteria and fungal, so the contribution for the dynamic is particularly on the environment factor. So because each of those we simulate the carbon assimilation and also carbon respiration, right? Then and also this is for the substrate environment factors. So temperature, moisture control of the growth size, growth rate, death rate, and also substitute how much carbon, how much DUC is available in the environment. So all those together to really control the carbon relative abundance of the bacteria and fungal. Yeah.
So do you see like, I don't know, characteristic latitudinal gradients in fungal to bacterial ratios, or like if you zoom out, I know most of the work has been like at particular sites, but if you zoom out, do you see interesting gradients or patterns?
I think yes. So this is the fungal biomass, bacterial biomass you see here, right? So the general pattern, we do have that, but exact value, we're still looking at the details values.
Cool.
Yeah. So I do want to emphasize once, this is represent the model simulator and also observation data. So this observation data is we collect a global kind of meta-analysis. Then we develop the global map of the bacterial and fungal biomass.
Great. Thanks so much. I'm guessing we should probably move on to our next speaker. Next up, we have Brett. There you are.
If I can share my screen here. Let me see. Hold on, having some issues sharing my screen.
I know it's always lots of fun when you're on the spot trying to get your screen up and running.
Filter. Okay. Can you see that yet?
Not yet.
Okay.
You might have to hit the share button. Sometimes I feel like I've selected it and then haven't hit the share button.
Yeah, it's bringing me into like some security and privacy. I don't think I presented on this computer on Zoom before.
Do you want me to go ahead and you
Keith
share
have your
your
presentation?
presentation with me?
He does, yeah.
Great.
Against the slides?
Yeah, let's just do that.
Thank you.
We'll give that a try. Thank you for sharing it with me.
Yeah, it's good to have a contingency plan. Thank you.
And
yep, that's it.
All right, go ahead.
All right, thank you. So just sharing some work about a data assimilation work that we're performing with CLM across the Western United States. This is a collaboration, or has been a collaboration with the University of Utah, and was funded from NASA's Carbon Monitoring System grant. And the timing is good. This was just accepted in the Journal James this week. Next slide. So why monitor carbon in the Western United States? And this has to do with the substantial carbon resources in the Western United States, but also the vulnerability of our forests, susceptible to forest fire, insect infestation, and drought mortality. And so I just put up the US Drought Monitor for last week. It's probably actually worse than this now because of the extreme heat. But this is a particular early season for exceptional drought to occur in the Western United States. And a little background, 2012 through 2015 was a particularly bad drought period that caused kind of large areas of mortality. And so it's a bit ominous to get this sort of drought this early on in the season. So there's the motivation for monitoring the carbon and the carbon flux. The challenge is that given the mountainous terrain, this is challenging to monitor with traditional carbon monitoring techniques like flux towers or atmospheric versions, and advance the slide. There we go. And so so it's difficult to do the traditional flux monitoring through top-down atmospheric inversions. And so what we're doing here is we're using the bottom-up modeling approach, using a CLM as a starting point, but then bringing in remotely sensed observations to help improve it. Next slide. It's a quick overview on the left. The cartoon is essentially showing how the assimilation setup works with Dart. And so we drive CLM, create 80 different instances of CLM through an 80-member ensemble of CAM4 Dart reanalysis. So 80 equally likely realizations of the atmosphere. And we use that to create the model spread. So to create that initial prior state in the cartoon right there. And so we bring in observations of above ground biomass, and leaf area. We bring that into the assimilation and create a new updated and improved posterior state. And I'm emphasizing the biomass and leaf area because we adjust those state variables within CLM specifically. We don't update all variables, but all variables are influenced due to the internal mechanisms within CLM. So soil carbon, soil water, and the fluxes are changing because we're updating the state. And so a more specific way to look at this on the right-hand side of the slide, going from the bottom up. So we're estimating the biomass observed prior state in the green text there, and we're getting that through a forward operator that assembles the estimated biomass by looking at the fundamental variables like live stem carbon, dead stem carbon, leaf carbon. It then compares that to the remotely sensed observed above ground biomass, and it creates an improved posterior. So the ensemble adjusted biomass to the right in the blue. And so that's the x-axis that creates an improved increment. And then we take that increment and regress that upon the other unobserved variables within the CLM model state. And so we take that and then we apply it to, and I'm showing a grid cell up there. So we apply all those updates to all the individual PFTs and columns in the restart file of CLM. So some pretty heavy lifting considering how complex CLM is. Next slide. And so just a little bit more about the methods. So we create a single instance spin up, kind of similar to what a lot of initialization to get the soil carbon and to get the initial biomass correct. We use a 1 degree grid for this. One of the unique things we probably do is to use the grid met forcing, which is specific for the Western United States, that removes a lot of biases in warm temperatures and low precipitation that you get for a lot of data atmospheres for the Western United States. And then we transition into the assimilation run. The time window is from 1998 to 2011. And we do something that we call looping, which means we cycle over the assimilation window 3 times in succession. So there's 3 kind of the red lines right there. And that it gets us from the free simulation using no observations and closer to the observations in a more gradual pattern. We also use adaptive inflation, which essentially is just a technique to make sure that we create or maintain the correct ensemble spread between all our different 80 realizations during the assimilation process to make sure that it doesn't collapse on us. Next slide. And so, as I mentioned before, we're using monthly above ground biomass observations and leaf area. I use the observation term loosely. These are really data products. So these are really like spectral reflectances that are then ground truth to get at a land surface property that we care about, like leaf area. We use an observation rejection threshold of 3 sigma such that we can remove observations that are too far removed from what CLM is telling us, and this is to protect against systematic biases that may be unaccounted for in the uncertainty. And then finally, we can localize both in space, so what realm of influence the observations have on the model, and also we localize in state space. So we can't adjust all 500 variables, all columns, all PFTs at once. So we have to make a choice of what is most related to dictating the carbon cycle. And so here I've just given some of the fundamental state variables that we adjust, both related to the carbon in the biomass and the nitrogen in the biomass. Next slide. And so finally to the results. And so when we include these remotely sensed observations within CLM, we show about a 30% reduction in the biomass in the leaf area with those plots above. And so you can see the leaf area is getting closer from the black from the free simulation to the blue observations, and the dynamic nature of it. The biomass takes a little bit longer and takes the 3 loop period before it gets more similar to the observations. You may note that the leaf area doesn't quite get the minimums, the seasonal minimums in the winter, and that's because those observations are rejected, and that's not necessarily a sign that the assimilation system isn't working. It's just protecting against potential systematically low observations of leaf area. And finally, the cumulative NEP in the middle of the screen, even though we're reducing dramatically the leaf area in the biomass, the main fluxes offset. So the cumulative NEP, or the net carbon uptake over the Western US, doesn't really change from the free run to the assimilated run. And so we show that in the bottom figure right there, the component fluxes are decreasing, but kind of offsetting so that the net carbon uptake isn't really changing. Next slide. And so just more diagnostics to understand how the assimilation system is working. What we typically do is look at acceptance rate. And so those are kind of the marks in the pink or the salmon colors. So at the top is the observations, the total observations possible, and then also shown in the observations assimilated. And the idea generally is to maximize the number of observations to assimilate, rejecting those that are just too far away. And looking at the root mean square error, obviously, is a key diagnostic to how well we're doing. Note that the leaf area RMSE is fairly steady throughout the assimilation period, where if you go to the bottom graph, the above ground biomass, it's definitely decreasing with time as more observations are assimilated. And so that's the direction we definitely want it to go. Next slide. So I've just shown a kind of like regional grid cell average behavior. So what are the individual PFTs doing? So I've just shown the major PFTs that make up the region between the temperate and boreal forests. And so we can show, as we would expect for leaf area in the top left there, we see reductions in the leaf area and also an increase in amplitude in the red and the assimilated lines as well. The biomass, as you would expect, is being taken out of the temperate and boreal evergreen forest as well. One thing we do notice is that although the natural vegetation types are fairly amenable to being adjusted, the crops are not. The crops generally, and I'm showing an example where it's not really budging at all, but generally they're more difficult to adjust. And this is presumably because of the way that crops are on tighter trajectories as far as the timing of their growth in senescence or harvest. And so we're thinking of ways to get around this. I'll talk about later. Next slide. And so how does CLM5 DART assimilation run? How does it compare against other estimates of carbon uptake in this area? And so I looked at, here I've plotted against FluxCom, which is a very different approach. It's a machine learning approach, uses flux tower data, and it trains other satellite data like leaf area data and meteorology. And it trains that to get the best kind of regional interpolated map of carbon uptake. And so what we can take from this is that looking at the seasonal average behavior, the ecosystem respiration between the two products is fairly similar. Where they start to diverge is the GPP in the top left there, which causes very different behavior in the NEP, both seasonally and cumulatively. And so you could see the machine learning approach is basically saying that this area is showing a lot of carbon uptake, where CLM5 DART is saying, no, it's a weak carbon uptake or more neutral. And that's regardless if it's the free run or the assimilated run. And so the reasons for this could be the way that disturbance history is accounted for. The machine learning approach doesn't necessarily account for like the site history or disturbance history that CLM does account for during kind of the spin up period. And this could indicate problems or motivation to adjust directly other variables within the CLM5 DART assimilation. So, but it's important here not to say like which one is correct. It's just kind of showing these kind of as bookend estimates of the carbon uptake. Next slide. So this is kind of the same figure, but then resolved spatially. So the free CLM, the left column, the assimilation, the middle column, and then finally FluxCom. And so you could see for the GPP and the ecosystem respiration, that flux, those component fluxes are pretty uniformly decreased from the free to the assimilated run of DART. If we go down to net carbon uptake, you can see that it's basically neutral. It's not showing, unlike FluxCom, that shows that trend of more uptake with elevation as you get into more forests. The assimilation is fairly neutral through the entire period. And I should say, this is 1998 through 2011. So the severe drought period from 2012 to 2015 hasn't come on quite yet. And so we're a bit skeptical of this, or at least wanted to probe this a little bit more. So we kind of looked at a little bit more diagnostics. Next slide. And so this is, of course, a water limited area, the Western United States. So I looked at this, one of the soil moisture limitation diagnostics in CLM5 with the highest water limitation with the darkest red colors. And as you might expect, it's mostly water limited within the Great Basin and to some extent the higher elevations. What we know for sure is that it's definitely influencing the spatial pattern of the GPP in terms of correlation. And so we know that it's having an impact. And one of the reasons why is that snow is definitely one of the key water cycling variables in the Western United States. So at least the modeled snow that we have, and I'm showing that in the bottom left, the spring snow water equivalent or SWE is biased low. It generally doesn't resolve kind of the southern periphery and western periphery of our domain, which could be a concern, and then motivates including more water cycling hydrological variables to our assimilation. Next slide. And so one more diagnostic that we're looking at, we also adjusted to look at the sensitivity of the assimilation to the various adjusted variables. So I show those kind of in the circled color boxes there from top to bottom, using a minimum amount of adjusted variables to more of a maximum amount of adjusted variables. And I show the adjusted variable behavior on the left column and kind of some of the other variables on the right. And you can see the adjusted variables are pretty consistent regardless of kind of the flavor of assimilation that I'm doing here. What really starts to vary are variables such as the soil carbon or root carbon, because they're not explicitly included in the adjusted state variables. And so this has an impact on the cumulative NEP on the bottom of the slide, showing some spread there, although it still indicates it's still a weak carbon sink. It does not agree with the strong uptake of the FluxCom estimate.
You've got 5 minutes.
Okay, cool. Next slide. So just reviewing some key points. So assimilating the observations of biomass and leaf area, although it does reduce the biomass and gives us what we think is a more reliable state of the system, it doesn't really change the carbon uptake. And furthermore, when we kind of probed some of the different assimilation settings, that kind of weak carbon uptake was fairly robust. Another estimate of carbon uptake, FluxCom, was much stronger in its regional carbon uptake. And we think that could be reasons due to the disturbance history, the methods of the different types of approaches, and kind of necessitating the need for including more water cycling variables within our CLM5 DART assimilation. Next slide. So where can we go from here? And so I talked about the water cycling variables. And so one of the cool things of the Western US is we do have SNOTEL sites that measure the snow water equivalent. So it is possible to use those as observations to directly update the snow layers in CLM and the different layers of soil water in the subsurface. There's been an explosion in solar-induced fluorescence measurements across the Western United States, both increasing temporal resolution and in time. And so those have a strong empirical relationship with GPP. And so we're including infrastructure in DART to be able to ingest those observations right now. Another approach we could take is really leveraging more high resolution land cover maps that give more metadata, more specificity to what the biomass observations are actually telling us. So it could allow us to map specific adjustments to the PFTs. And so getting back to the crop PFT, where we couldn't get it adjusted, if we could apply a smarter forward operator, we could get more specificity in our adjustment directly to the crop. And then next, the finer spatial resolution. So you might be asking, like, well, why don't you just go to a super fine spatial resolution? It is true in the atmosphere. So in this work, we use the CAM4 reanalysis, which is fairly coarse, 2x2. Now available is a CAM6 reanalysis, which is a 1 degree, which would be better to use. We are using a 1x1 land surface grid as well. But what we found in work that, and I don't have time to show now, is once you get below 1 degree resolution, it doesn't really give you much improvement in the carbon dynamics. And that could be because, we're missing fundamental representation that could be represented in the hill slope hydrology model. And also both in the hill slope hydrology and then also, I forgot what I was going to say, but and also parameter estimation. And so we've also shown that it could be more important instead of spatial physical resolution, ecological resolution, meaning better plant functional type representation is still important in this area. And so that brings to the last point on the bottom right that I've talked about the data assimilation research, the ensemble common filter in terms of state adjustments, so things like biomass and leaf area, but it is possible to use it in kind of a parameter estimation mode where it kind of has a more like a stronger impression on the model, where if you update the state, it's fine as long as you still have data to keep on updating it, but eventually the model could lose memory of it, and so a more kind of fixed improvement could be through a parameter estimation approach. Next slide. So that's cool.
You've got like 30 seconds left.
Sure, perfect. Yeah, this slide got a little messed up, but anyway, this is just to show all the different models that we support, not just CLM 5. And here are the links to the documentation. We've just kind of refurbished our documentation for Dart. And so please take a look if you have any questions, and thank you.
Great, thanks so much. I think we should move on to the next talk, but I'll note that there were a couple of questions in the chat. So if you want to have a look at those about crops and Snowtell, and one more I haven't had a chance to read yet. So go ahead and continue that conversation. Next up, we have Nicola, and I hope I pronounced that correctly.
Hi, let me just get this up and running. Okay, are you seeing?
So we're actually seeing your presenter mode. Can you switch the screens? Yeah.
Yeah. There we go. Good, right?
Yep.
Awesome.
Great. Okay, take it away.
Awesome. Hi, everyone. My name is Nicola Wiseman. I'm a PhD candidate at UC Irvine, and so today we're going to take a little bit of a deviation from some of the land and lake talks we've had so far, and I'm going to talk about our ocean BGC in CESM. And so what I'm going to focus on here is the biogeochemical impacts of plankton stoichiometry in the CESM. So this is some work that I've been doing over the past couple of years with Keith Moore and Adam Martini at UC Irvine, as well as Ben Twining at the Bigelow Marine Lab. So, I feel like this is exactly what this group is really focused on, but one of the ocean plays, a really key role in carbon storage, as well as our the ocean and land both play key roles in carbon storage, with about a half of carbon emissions being left in the atmosphere, 25% going up in the land, 25% going up in the ocean. And the uptake by the ocean is really driven by both our solubility and our biological pumps. And so for our biological pump, it's really driven by our phytoplankton. And so phytoplankton, they take up DIC from the surface waters as well as nutrients and use it to incorporate into their biomass. And what this is really controlled by, in this productivity in general is really controlled by, is the nutrient availability. So this is a figure from a paper by CM Moore et al. 2013, showing bottle experiments of nutrient limitation throughout a decent portion of the global ocean, where we have red is iron limitation, green is nitrogen limitation, and then our black circle outlining is our P co-limitation. So with the background here being dissolved nitrate, and then in the panel B being dissolved phosphate. And so what we really see here is we see the elevate, our HNLC regions, our high nutrient low chlorophyll regions, where this iron is limiting in our North Pacific, Equatorial Pacific, Southern Ocean. We see our phosphorus and nitrogen co-limitation in the Atlantic, subtropical dryers. And then we also, I don't have it listed in the key here, but we also have SI limitation as well in some of our upper ocean or our high latitude regions. It was really driven by sort of the bloom cycle of our phytoplankton. So one of the things that we're able to do with this information is we know that variable stoichiometry within our phytoplankton modifies the carbon export, surface nutrients, and the spatial patterns of our nutrient limitation. So we have our primary productivity by our phytoplankton photosynthesis taking up, I'm specifically going to be focusing on nitrogen, phosphorus, and iron here. They take up these nutrients as well as carbon to build their biomass. And then the ratio of those nutrients is then can be exported into the deep ocean, this is what we really care about when we're talking about oceans and biogeochemistry, is this carbon export from the surface layer and that long-term storage, and sort of getting an idea of the role of how that may change sort of in a future climate scenario. So within the CESM model, I'm working with focusing on that region here. I'll get to that in 1 second. Yeah, so here, due to some recent expansion of methods in determining iron to carbon ratios of our phytoplankton, we've actually, the database on these specific cellular measurements has really been expanding. This is thanks to work by Ben Twining and collaborators, where we're able to measure cell-specific iron to carbon ratios instead of just bulk POM iron to carbon, which has been done in the past. And so this is a collection of measurements that have made in the past couple decades of iron to carbon. And one thing that we really see here is that these ratios can vary more than tenfold. So, our lowest measurements are down in our single digits, but we also have, in our high iron regions, like our the Saharan dust plume and our coastal regions, you can have very highly elevated iron to carbon ratios, upwards of 60 to 90 micromole per mole here, even further above that, depending on the organism and depending on the conditions. So we get our lowest values in our iron limited regions, our HNLCs, that high latitude North Pacific, equatorial Pacific, Southern Ocean. And then again, we see the more elevated values in those dust plumes as well as in our coastal regions. So this is, a data set that is being expanded slowly but surely. But one of the things that we've been able to look at with this, data set of iron to carbon is the relationship between cellular iron to carbon and the local dissolved iron. So this is a plot. This is just from the observations that have been gathered. We've got this linear relationship that really becomes pretty clear with iron to carbon versus dissolved iron at those locations. So there's this correlation between the two, and it's consistent with this idea of frugality, where, in our low iron conditions, organisms are able to reduce their cellular iron quotas, their cellular iron uptake. And then when iron is in surplus, we have this luxury uptake that is able to occur. This follows along with some of the work that has been done by the Martini group at UC Irvine, looking at phytoplankton stoichiometry, specifically C to N to P, and POM stoichiometry as well. But so this is from a 2015 paper where these dots here are showing, or these sort of blocks are showing field observations, variability. And, we have P to C versus PO4 concentration, N to C versus NO3 concentration. And so there is a lot more variability in our P to C ratios versus our N to C ratios. But one thing that is really key is that in the sort of low nitrate regions, or when nitrate is very low and nitrate's limiting, there is a lot more variation that you sort of don't see along the line. And nitrate is one of the most limiting nutrients in the ocean. So this is sort of a region that we really do care about, is these low nitrate values where N to C may be more variable. So for this study, what we did is we compared two CESM BEC simulations, one with fixed stoichiometry and one with variable stoichiometry. So for our fixed stoichiometry run, we used Redfield C to N to P, so that's a 120 to 16 to 1. We used a fixed iron to carbon ratio of 10 micromole per mole and a fixed SI to C of 0.133. This is an SI to N of 1. And then in our variable run, we have C to P varying about 100 to 200. N to P is also variable, but these two are linked by a constant C to N ratio of 120 to 16. So it's the P that's really varying here, even though we have variable N to P here. It's the P that's changing, and N is fixed. So for our iron to carbon, we have a range of, for our 3 phytoplankton groups, we have our small phytoplankton, our diatoms, and our diazotrophs. For our small phytoplankton and our diatoms, we have a range of 3 to 90 micromole per mole. And then for our diazotrophs, we have a range of 6 to 80, as diazotrophs have higher iron requirements due to the metalloproteins that are found within them in order to undergo nitrogen fixation. And then we also have our variable SI to C for our diatom group. And so these are 300-year runs. Those are 2 300-year simulations where we analyzed the last 20 years. So the way the variable stoichiometry is formulated in the BEC is sort of this nutrient to carbon ratio as a function of nutrient concentration. So here we have our nutrient X. We're just generalizing with being our phosphorus, iron, or silica. We have our maximum nutrient to carbon. This is that we prescribe. And then we have this optimal nutrient concentration for growth. And so when the ambient concentration falls below this, the phytoplankton will reduce their quotas accordingly in order to sort of account for those lower nutrient conditions until they reach a prescribed minimum. And so this applies for both the P, the phosphorus, iron, and silica. And so overall, we end up with the ambient concentration determining the ratio for new growth. And as I said before, the nitrogen to carbon ratio is fixed at 16 to 120, where the uptake is dependent on the available nitrate and ammonium. So with including these 3 variable nutrients, I'm going to focus on iron really quick for a second. So with this new formulation of this wider iron range of roughly 3 to 90, CSM is able to better capture the iron to carbon ratios that we measure in observations. So this is just showing the iron to carbon and dissolved iron from the model extracted at the same locations as the observations. And so we see that the simulated iron to carbons ratios vary widely, and they have a direct impact on the carbon export by the biota. So here we have this is going into those 2 runs that I was just talking about. So we have the variable stoichiometry run, fixed stoichiometry run, and then the difference between the 2 versus our observations. So as a result of increasing the variable, or including variable stoichiometry in the model, it modifies the efficiency of export by the biota and therefore the surface nutrient distribution. So here we have the iron, and the main thing that we see here is that in the variable model, you have a lot more iron uptake in our coastal regions, and so therefore lower surface iron compared to our fixed stoichiometry run. And then here, this is a lot of plots to look at, but we have our nitrate, phosphate, silica. And so again, we have the variable run at the top, fixed in the middle, and then the difference. And then I've included Worlow's analysis here at the bottom for the nitrate and the phosphate. And the main takeaway here is in these difference plots, which just shows that the HNLCs expand significantly in our fixed runs due to widespread iron limitation. So the iron to carbon that we have prescribed of 10 is too high there, leading to an excess of iron export from the surface layer and buildup in the intermediate layers, and therefore increased iron limitation. We also get a reduction of surface phosphate in the subtropical gyres here in the fixed run. And this is due to the C to P ratio being too low or P to C being too high. So again, we're having too efficient export of phosphorus in regions that where phytoplankton would in the variable run be reducing their quotas accordingly, therefore increasing their C to P. And our SI as well sort of follows that same pattern as our phosphate. So these are maps of growth limitation by nutrients. So purple is nitrogen, blue is iron, green is phosphorus, yellow is silicon. So in our variable versus our fixed run. And if I just sort of focus on, our diatoms here, in general, we do see a major expansion of these iron limited regions. And the HNLCs in particular. There's also a major expansion of P limitation, like I said before, due to the increased export of nutrients. And we also see expansion of SI limitation in the high northern latitudes. So one of the things with variable stoichiometry, including this in the model, is that it really acts to reduce the export efficiency and therefore retaining limiting nutrients in the water and allowing for further recycling of them and fueling more productivity. So this is our surface. Okay. This is our surface chlorophyll in our two runs. And then again, our difference overall, lowering the variable nutrient quotas where they are able to sort of lower their nutrient quotas accordingly in regions where they're limited, by phosphorus or iron, is able to maximize their efficiency. And so you get, these greens and reds here in this bottom plot. We're getting more chlorophyll in our variable run because they're able to sort of utilize and recycle these nutrients. And then we have our particulate fluxes, again, variable, fixed, and then the difference. And in general, overall, we see that export is pretty much except for in this sort of western region. Variable stoichiometry is maximizing this use of nutrients, leading to increases in productivity and carbon export relative to the fixed run. And so even after 300 years, the POC export is converging a tiny bit, but it would take, I think, a couple thousands of years in order to get these to actually converge. So as soon as you sort of introduce this variable stoichiometry, it really has a first order impact on the carbon cycle. This is just air sea flux. And I'm actually going to skip sort of to our summary really quick. So again, we have these first order impacts on carbon cycle by including variable stoichiometry that is modifying the biological carbon pump and our air sea CO2 fluxes. Our variable stoichiometry strongly impacts surface nutrient distributions and patterns of limitation. And accounting for this variable stoichiometry it's critical to projecting the ocean biogeochemical response to ongoing global warming. And our next step is to include variable N to C, which is currently fixed, but there is some variation as evident from observations, especially in our oligotrophic gyres. And so it'll be very interesting to see what accounts for what happens with this as we go to including this variability as well. Thank you.
Great, thanks. We have a couple of questions already. I saw one hand that went up and then went down. So if you still want to ask your question, and also some clapping. I'm going to go with Nikki's question in the chat first. Which chlorophyll map looks more like the satellite observations?
That is a great question. I should have included that one in here. It's definitely the variable. The variable map is much closer to matching satellite observations, because especially with the fixed run, you really get a major underestimate in the equatorial Pacific upwelling region of chlorophyll, as well as there's some dampening of the bloom cycle in the high latitudes as well.
Great. Okay, Keith, do you want to go ahead?
Sure. Thanks, Nicola, for that great presentation. A lot of the story you were explaining in context of the iron limitation, say in the equatorial Pacific, in terms of the impact on the HNLC. Have you done or considered doing experiments where you don't do all of these variable stoichiometries, but do them one at a time?
So I do have runs that I will be doing soon where I'm just turning on and off the iron. So I do have results from that, and I don't know if we have one with just turning off the P or the SI, but would be easy to do so. And that might be something that could make, as I will be doing sort of a full look with the variable versus fixed in sort of a long-term climate simulation down the road. And so that would definitely be something that could potentially tell an interesting story as part of that project.
Great, thanks.
Cheryl, I think you had your hand up earlier, and then it disappeared. I'm sorry, I missed whose hand that was, but go ahead.
Well, I have a bunch of questions. One of them got answered. So my understanding from this is that the export ratio doesn't change, but the amount of productivity changes because night better utilized. Is that correct?
Oh, that's a great question.
Like, is there more ballasting if you have more iron? Like, I understand if like the plankton has more iron and it's exported, then it's taking more iron out of the surface into the deep ocean. But like, does it? So did you change the export module?
No.
Okay, that was my question. As far as
did not change export at all. It's fully just being driven by the phytoplankton changes in these versions.
So that was my basic question, and then my other big question, and I've often thought this about this model. We have these super plankton, plankton functional types, right?
Mm-hmm.
And so, in reality, there's a great diversity of diatom species that do different things with their stoichiometry. And so, you showed some really nice plots of matching the observations, but is there a limit to plankton functional types for this? Do we get to the place where we need to add more or have a different paradigm of how we do it?
So that is something that actually is being worked on by one of the other students in my group. We are working on I think it's a 7 phytoplankton model, 7. I could count them, but that is something that's being worked on in conjunction with some of the scientists at NCAR in our group. So with Marble, the sort of new biogeochemical model, one of the great things about Marble is it's modular. And so you're able to add and remove phytoplankton groups as you see fit. So I think it's 8. I think it's 8 phytoplankton. There's an 8 phytoplankton model that is currently being worked on, and they will have, these individual sort of variable stoichiometries. So I think it'll be interesting to see, one of the issues with doing that though is then it's more knobs to turn. And so it can get very difficult to sort of narrow down what we really think is correct. And a lot of that is limited by observations, and whether or not we have sort of, specific species stoichiometry observations. And so I'm always a proponent, more studies, more field work, and then we'll throw that into the model as we get it.
Keith, do you have a really urgent question, or should we move on to the discussion?
It was a brief follow-up to
Okay.
to the first question from Cheryl. So I don't think you showed community composition, Nicola, but I would have guessed that if community composition, if there is a change in export ratio, that could have come about from a change in community composition. And so it's possible that the ratio changed if there is a shift in community composition from these changes, but I'm not sure that your plots showed that or not.
Yeah, I do have them. If you send me an email, I can absolutely find them and show them to you, but I just don't have them in this specific presentation. But I'm pretty sure there are definite shifts.
It would be really interesting.
Small dominated.
This is great discussion, but I think we really have to get going, move on to if we want to have the full group discussion about the priorities for the working group, I think we need to move on to that. Sorry.
Thanks for
fine.
keeping
Thank
us
you.
on time, Gretchen.
Yes, thank you, Gretchen.
Thanks, Nicola, for your talk and for the good discussion. Okay, next, as Keith mentioned earlier, we were hoping to have a group discussion, and I don't know if Keith wants to try to pull that slide up right now, but we are going to send people into breakout groups so we could hopefully have some conversations in slightly smaller groups about what the working group might do, as I'm trying to paste the link to our slides in there, what the working group might want to work on in the future and prioritize, and how we could coordinate those activities to bring people together around this topic. So this is a Google slide deck where we can take some notes. We can share this link also within the breakout groups. And then I think we're planning to head into probably 3 breakout groups. And I don't know if Todd was going to set those up or if I should set them up.
They're already set up, Abby.
Great. Okay. Thanks.
Is it going to be 15 minutes long?
Yeah, I think let's take 15 minutes in our groups, and then we'll come back and have a full group discussion about what we talked about in those smaller groups. So thank you to all the speakers, and we hope that many of you can stick around for these conversations as well.
Okay, we're ready to go?
Yeah.
Okay, here we go. Are you all able to press the breakout rooms button and join a room? Okay. Set the alarm for 14 minutes. We are currently in breakout rooms. If anyone is still here, you can press the breakout rooms button and join a room.
All right, welcome back, everyone. Is it okay if we take down the agenda for a moment here so we can see each other?
Someone else put that up. Let me see if I can remove that. Ryan, are you there? He must have stepped away.
I'll try, hold on.
Funny, dude.
All right, I took over the screen sharing and then turned it off. Okay, well, I hope that people were able to have some conversations in their groups. I thought we could share out some of what we talked about in the different groups, but I'd be happy for it not to be me talking. So does anybody want to volunteer something that came up in their group that they wanted to share? And I'll keep taking some notes. So I would say just unmute yourself and go for it. Go for it, Will.
I'll go for it. The Dave asked the provocative question, as is his MO during the meeting, which was like, does the biogeochemistry working group need to exist? And we came up with some. Well, it's not really doing any model development, and so if it does need to exist, it needs to exist kind of more for scientific questions, maybe more similar to what the Paleo Climate Working Group does. And it also needs to exist in some form because it has a really nice computing allocation, which is useful for the development of biogeochemistry. So even though the working group itself isn't doing the development, its computing allocation is very useful.
Because the runs are really expensive.
Right.
Yeah. Okay.
Yeah. There was more to it that's on our sheet, but that was the most interesting, I think, part of the conversation was like a bit of soul searching about whether it's necessary.
I would say that in answer to, sort of the need for science questions, that was, I thought, where our group spent most of our time, was thinking about whether there were questions that we could answer about how the coupled climate system would respond to negative emissions, and whether we can make a constrained model prediction to understand, 10 years from now, given a certain level of atmospheric CO2, to what extent is it because folks did not meet their emissions production contributions, or the extent to which natural sinks might have changed. I think those are a couple of really interesting topics that, as something else Gordon noted, we want to really put BGC and carbon at the forefront. So we want to ask provocative questions that will get other aspects of CESM interested in what we're doing.
Do people have more ideas about science questions that they thought were really well suited for the BGC working group?
This is kind of related, but to what Gretchen was just talking about. But I think it's important to recognize that to try and understand the carbon cycle, you have to actually be able to measure the fluxes of carbon, and we can't actually do that right now at large, continental scales. And we can't do that because the atmospheric CO2 inversion techniques essentially don't work. But if they did work, they don't have the spatial resolution or the process information that would actually allow you to really understand what was going on. The Earth system models are the key to understanding the processes that are driving things, yet they aren't generally used for flux estimation. So I see a big, gap, but possible opportunity for interconnecting these two sort of fields of study to try and use Earth system models in a flux estimation framework. Brett's talk was starting down this direction. I thought that was really exciting. A lot of hurdles, but I think the process information in Earth system models could really be brought to bear in ways that hasn't previously.
So I was wondering if that gets to, now I'm forgetting, it was maybe Will said that stated that the Biogeochemistry Working Group doesn't do any model development. But that's a question of if we just don't do it right now, it certainly has in the past, and is that something that it needs to do in the future? Like if there was to be if we were to create the infrastructure for what something like Britt is talking about, does that fall within the Biogeochemistry Working Group, or is that something that gets done elsewhere? Or does it matter? You folks tell me what's important about that.
One thing that I didn't mention in our group because I had to go briefly, but what's neat about biogeochemistry is that we have these new observational systems that are coming online. Like in the physical realm, observational systems have existed for quite some time and they really haven't evolved, but we have new observations, in particular in ocean biogeochemistry. We have biogeochemical Argo measuring nitrogen in the ocean, measuring PCO2 and pH in the ocean. And CESM could be a test bed for biogeochemical OSSI type experiments in a way that no other model has really gone. No one else has really gone for that yet. So, and probably there are similar things you could think of for the terrestrial carbon cycle as well, where the BGC component of the land model could be used for an OSSI type test.
I don't know, any of the land model folks want to comment on that? David, Gordon, Will, probably, or Danica, other people are probably here. I only see a few videos.
What's an OSSI test, Nikki?
An observing system simulation experiment, right? So how many biogeochemical Argo floats, a very simple one, how many floats do you need to capture the large scale pattern? Well, you don't really know because you haven't put any out there yet. So in the real world, so why don't you put them into your model and let them advect ground with the Lagrangian flow and subsample the model biogeochemistry at certain locations, and then you can get a sense of whether you can back out the large scale patterns or the small scale patterns or what have you.
Yeah, Abby, I'll just add to this. Again, historically, we took the model development out of BGC and put it back into the ocean and the land because that model development was being done independent of the ocean model, the land model, and that was causing problems. So we sort of felt like the development had to be in those working groups. But that said, yes, there was a huge need for the biogeochemistry working group. And I think maybe the need is sort of what Nikki has been talking about in terms of community experiments, and even what Britt's talking about in terms of sort of, can we actually get flux estimates out of an Earth system model now? We need to think of a community project that would be driven by the biogeochemistry working group that would evolve development, but it'd be development in terms of creating like this whole sort of carbon prediction system, for example. Can we actually do something like that? That would touch into other aspects because there's the Earth System Prediction Working Group. There's the large ensemble activities going on with other working groups. But again, we can create sort of like a big activity that's led by the Biogeochemistry Working Group that spans across all aspects of CESM. And that's the need, and that's the rationale for the BGC Working Group. I kind of like what Britt says because it puts another value to both the land model and the ocean model, not just providing sort of surface fluxes to the atmosphere or looking at impacts of climate change, but actually sort of becoming a really important scientific tool. I think what Brett was talking about today was actually really kind of interesting, of comparing his assimilated product to FluxCon. That to me seemed like kind of interesting, right? There's a lot going on right there to explore.
I think I really like this idea, Gordon, of a large-scale unifying project. I have heard from some corners some pushback about making sure that you balance that with bottom-up PI work. And so we need to make sure that we still have that we're welcoming to PI initiated, what we used to call entrepreneurial science, with the large scale projects. And so I think there's a balance to be struck there.
Doesn't seem too hard, right, to balance that. If we're not shutting, there's no intention to shut down PI driven work. So I don't think that's that much of a tension, right?
Okay.
It probably works the other way, that if you do have some sort of an overarching mission, then PIs kind of like latch onto that and find niches that are related to it.
That can be a good counter argument.
Yeah, so just to add to that, Keith, in our group, we talked about how there needs to be more people who know how to do what you do and run these like coupled climate simulations. And that one way to do that is have PIs outside of NCAR get funding for postdocs and grad students who could learn those skills. So we could, take that out of NCAR and spread it around more.
Thumbs up.
Yeah, I think that having, there's a lot of interest from the outside community, and at least in talking to me, for having fully coupled simulations. And I think having projects that are if we target a few of these larger group projects within this group and provide those simulations for analysis with the fully coupled simulations, that could provide another tool. And then you're right, then postdocs and other people have capability of analyzing those. I realize that some of those are available through CMIP and also through the Lens 2 work, but we could potentially provide additional simulations. The other thing that I wanted to mention is just that we focused a lot, and I think this group still focuses a lot on CO2, but nitrogen is also really important for reactive chemistry in the atmosphere, for example, or for leaching. And so that's an important component. And it was really interesting to see all the talks on methane and thinking about including methane in some of these projects.
Right first, then Dave.
I was just going to say, we talked about this a little bit too, and these are sort of these intractable problems that we always talk about and never make progress on: closing the nitrogen cycle, closing the methane cycle, or doing prognostic. And maybe the working group can be to initiate this, but I was suggesting maybe it doesn't need to. But now I'm wondering if that was the right way to think about it, is to say the working group needs to look for opportunities for a funded proposal that would involve multiple working groups to do this, because it's like, to really do the methane cycle, it needs everybody to be involved, right? So if the land model working group submits a proposal or somebody in land, it's never going to be enough, right, to do the whole thing. So either it needs to be base funded activity, or it needs to be some sort of really integrating. And that would be another type of activity. Maybe there's the simulations or the sort of activity that the kind of activity Britt was talking about, but also another would be a little bit model development, more like integrating across components, right? Model development. So.
But even for carbon, we're still missing some big pieces of the integration, right? Especially if we're thinking about estimating carbon fluxes. It seems like there's some major infrastructure pieces that are missing there. And we talked in our group about riverine inputs to the ocean from land. But as far as I know, is something that's always on the list for this group, and we never have made progress on. I guess it could still be thought of in their priority with how they impact these bigger goals. Sorry, go ahead, Britt.
I think you're right that if you tried to tweak the parameters in CLM and Marble to try and match atmospheric CO2 exactly, you would run into structural problems eventually. But you may have so much tunability that you could actually match the real atmospheric CO2 without necessarily getting it right for the right reason. but I'm looking forward to, the PPE, I think that's the right acronym, from the CLM parameter ensemble, and just to see how much how much can you swing, say, the seasonal cycle amplitude or the atmospheric growth rate just by selecting parameters within a reasonable range. And I talked to Matt a little bit about the limitations of doing something similar with the ocean model. I think, if I understand correctly, it has to do with the sort of the timescales of in the way that the physics in the ocean sort of sets the stage for the biogeochemistry. But he had an idea for doing something where you were nudging the deep ocean and just doing your parameter ensemble on things that mattered for the near surface layer. So it may be that once we have some experience looking at PPE that we find out there is a way that you could team up with the folks in DART. I mean, NCAR has these big efforts in data assimilation that haven't really been focused on CESM specifically. It's my impression. So there's also an opportunity there.
Yeah. Well, with Brett moving to NCAR, that helps, at least from the land side, right? He's got tons of land experience and really interested in land data assimilation. So it could really, plus Andy Fox still being around, we're kind of getting more of a critical mass on land data assimilation.
And we've got Dan Manrine in the oceanography, who also works with the DART group. So we've started some conversations about, but they've kind of stagnated in recent months. But we are trying to move forward with trying to have more interaction with the DART group for Ocean BGC. And I would love to be able to take lessons learned from CLM PPE and apply them to Marble PPE.
I will point out there is a group that did a couple of carbon cycle perturbed physics ensemble. It's out there. It's like a 2012 paper on the UK model.
Okay.
They got like 300 ppm spread in the atmosphere across all of their parameters. It's kind of crazy. Sorry. Anyway, other comments?
I have a
share
comment.
a link.
I have a comment about Keith's opening presentation for this subgroup meeting, which was that he said, "We've noticed there have been fewer presentations submitted." And I just want to say that that's partially my fault, and I apologize, but I didn't have the bandwidth to have myself or anybody in my group submit presentations either this year or last year because of the pandemic. And I suspect that that's true in many of the working groups. So I wouldn't use the statistics of the previous two meetings to characterize interest in ocean biogeochemistry or the biogeochemistry working group.
Thank you. No apologies necessary.
Anyone want
to? And thank you to Nicola for representing
Yes,
Irvine.
I'm really glad we had some ocean representation. Thank you.
Of course, happy to do so.
Does anybody else want to comment who hasn't had a chance to yet?
Forrest, drop some wisdom on us.
Yeah, I see. Or Jinji, any perspectives from someone who hasn't been part of been hearing the same working group discussions for a long time?
Yeah, I'm really new to this community, so as I talk, so for people who don't know me, I'm Junji Liu from GPL. Primarily working on the top-down flux immersion and the carbon climate feedbacks. So I think listening to all these talks is very interesting, but I think it would be great if the modeling community have more interactions with the observation community to incorporate more observations into the models. I think Brett's talk trying to use DART system to optimize the state vectors are very interesting. I think what Brett suggested of using CLM to use to estimate surface, basically estimate fluxes, is also very interesting. But I want to say that top-down flux immersion is not, still have some usage, we probably could try to leverage both assets, try to understand the internal variability, especially, and in order to do the better prediction in the future. And for the community project, it would be interesting to at least to simulate the past internal variability from the observation, and then we think about the future predictions.
I was actually wondering if Gordon or Nikki had any thoughts on that, because whenever I think about this, I think about your paper where you show that we can't actually constrain the future carbon cycle by using the past observations. Do you think that there are any aspects of carbon cycling that would be more useful for constraining the future, or is all hope lost? The paper was obviously very influential in my thinking there.
Yeah,
I'll let
but
Nikki
in a very
start with-
negative way. No, I mean, I think certainly we would do better to improve our model observation match based on the few observations that we have. But I think we can also use our model to promote the collection of additional observational based data. So I think we can use it in reverse to argue for why we needed more in the past and we didn't have it, and what we can be doing moving forward.
I've always wondered whether or not, you know, your conclusion is just that we didn't apply enough constraints, right? So well, I think one thing I'm hoping that happens in the PPE is we're going to try to apply a lot of constraints to even to things that aren't directly the carbon cycle, but maybe are going to be related, you know, to water and whatever, and try again and see if with a much richer set of constraints there is actually. But just to be a little more optimistic, that maybe there is a chance.
Well, somebody's got to go negative here, so I'll do that. Which is, you know, I do think we have to. I agree. You know, more data will help solve the problem, but will it actually? It will help, but it won't actually, you know, eliminate the problem. And I don't know. I still have this view that we have to live in this world of uncertainty. That if you just look at the diversity of life in the world, there's no way we can ever truly capture the difference, all the different life forms and organisms, and even just the spatial variability of ecosystems. And so maybe we have to start living in this world of uncertainty. I don't know what that means. You know, I know it's perceived as being negative, that we should actually be converging on an answer, but I have this view that maybe there are multiple answers out there. And we probably need to start living in that world of multiple answers. And so those are some of the things that Nikki and I were trying to get at, you know, with what we were doing. But what I also want to end on something is optimistic, which is if you look at the other paper, that Nikki led on carbon cycle prediction, there is some predictability in the carbon cycle. So despite all this uncertainty and all this stuff, we actually can do some things that are going to be really, I think, helpful in terms of looking at how the carbon cycle is actually evolving in the future, and maybe sort of how we want to, why it's changing, and what we could actually do to sort of make predictions about how it will change over the next year or so. So yeah, this is the world I live in now. There's a pessimistic side of me and there's an optimistic side of me, and they sort of keep fighting among each other. So maybe I'm going, bipolar. Yeah.
One of the things that Jerry Meal has been struck by in this predictability work is that we often find in the biogeochemical world higher or longer lasting predictability than you do for physical variables. And he has hooked onto that. So we have sold a physical climate scientist on biogeochemistry through these prediction simulations, I think. There's more that we can do like that, I think, as biogeochemists.
I can think of a land example of that too, potentially anyway, that you might be, knowing something about the vegetation and what you expect it to do is going to tell you something about what you expect in the physical climate system. And that may have a longer term memory associated with it than what's influencing the physical climate system. Well, we've already held everybody quite over, but this has been a fun conversation. So thanks everybody for participating. Are there any other thoughts that Keith or Gretchen, you wanted to make sure we captured?
I think Gretchen needed to sign off.
Okay. It's late there. Yeah.
Thank you everyone for participating in this conversation. This is really useful to hear these different perspectives and input.
Oh, one last thing, Keith, can you paste the link to sign up for the email list, which we should have done earlier also, into the chat?
Let me find it, yeah.
We discovered that we have an email list, but probably nobody's on it. Like, I'm not sure I'm on it. So we were hoping that we could try to get, we could share this around and try to get people who are interested to sign up so that we could at least have a hope of contacting people and coordinating some of these ideas going forward. At least to, you know, it's obviously like as people are interested, but we didn't otherwise know how to contact the people who were interested.
Is there any hope of having an in-person meeting anytime soon?
Someone else has to answer that question.
I'm not sure what timeline that's going to be on NCAR property.
It's looking like maybe next year for a hybrid, but don't take my word for it because things are still being worked out.
I miss Breckenridge.
Well, yeah, off-site might actually be a lot easier. I think there's the tea leaves that I've been reading are that, you know, a large actual workshop won't happen until 2022, but that there might be an opening up of having like a 10-person meeting sometime this fall. But yeah, if somebody wants to organize something up in Breckenridge.
I think the facility decided not to rebuild after they had their roof collapse.
They could make more money off of condos.
They didn't. That was my understanding, is that their roof collapsed because of snow, and I think they decided to eventually. They didn't rebuild the conference facilities.
Maybe a little off topic, because presumably there are other places one could meet. I know everyone loves Breckenridge, but mostly it'd be nice to see all of you.
It would be nice to be in person, that's for sure.
One thing that I was just thinking is I wonder if this group would be interested in an Aspen Global Change Institute meeting, when they're meeting in the mountains.
That'd be awesome.
Have to focus on some topic beyond just biogeochemistry in the Earth system, but yeah, I think that could be a really interesting topic.
But maybe focused on one of these science ideas.
Yeah.
Specifically.
Yeah, exactly.
Irreducible uncertainty in the carbon cycle. Living with uncertainty in the carbon cycle.
Yeah, well, thanks everyone for participating. Really appreciate it.
Yes, thank you very much, everyone. And we hope to see you soon again. And yes, as Nikki says, in person would be great too.
People can stay in chat here too. I think they told us we obviously went well over the time, so we don't have to leave immediately. But
Ryan, how long do we have?
What, to chat?
Yeah.
Feel free to keep on going. I mean.
Okay. Thank you.
Yeah.
I'm going to sign off. Thanks, everyone.
We'll
Thanks
stay
for coming,
around
Britt.
more,
Thanks, Britt. Thanks for your input.
See you around.
Hi, Junji. Oh. Everyone's leaving anyway. That's fine.
Boris, how have you been?
Okay, I have to apologize that that I had to start driving home during the breakout.
Oh, okay. No need to apologize.
And that's why I was breaking it. I like this discussion. Some big, complex, chemistry question that involves lots of groups potentially, and some. So you know, one thing that's really starting to get a lot of attention on the DOE side is coastal systems and the carbon cycle. We do get hooked up for those for riverine runoff. If there's some interesting questions that we could ask that would, you know, entrain a larger body of people out of CESM.
So you're saying that they might be doing that riverine flux development?
Yes.